Hi, everybody. It's sharp 2 a.m., 2 p.m., sorry. Maybe 2 a.m. for some of you around the world. So I'm very happy to open the first session of the third day of this meeting and the last day. Just a reminder for the speakers, you will hear a bell ringing 2 minutes before your time is off. So please wrap up your story and then we have some time for questions. Okay, the first speaker of this session will be Lois Weissman. She will talk about the regulation of a receptor recycling pathway via a phosphoinositate cascade. Lois, the screen is all yours.
Okay. Well, first of all, Lucio, thank you so much for inviting me to be at this conference and for having the courage to make it a hybrid meeting so that those of us who are not in Italy can attend. And Vitas, as I said, I'm so jealous that you're there. I admire that, and hopefully I can see everyone next year. So I'm going to share my screen. I don't know why this is not working. I'm so sorry. It worked a few minutes ago. Okay.
It's okay now.
Yeah. Okay, great. Okay. So are you seeing just my slides?
Yes.
Great, because I have a second monitor set up. Okay. So my lab has had a long-standing interest in how events are organized in the cell in space and time. And this is a process that's important no matter what system or organism you're working on. If you go down to the individual cells, every cell type is organized differently. The organelles are different places in the cell, and things are moved around in the cell during differentiation and during other types of signaling. And so how does all of this occur? And we can study this kind of process, not just in these beautiful mammalian cells. So here a polarized epithelial cell, and here is a primary cultured hippocampal neuron. But we can also study this in yeast. And here this is a budding yeast. And the mother cell is actually very different from the bud. So we can even study this here. And several years ago, we realized that there was a process in yeast that was very dynamic that we could study. And this was inheritance of the yeast vacuole or lysosome. And I just want to show you this movie because I love it. So these are still images of what you're about to see in the movie. So the green over here, this is the lysosome or vacuole. And over here, this is the mother cell, and this is the bud. And you're going to see a little dancing around here. And then suddenly this projection is going to shoot out and go directly to the bud. So here's the movie. So we see this kind of dancing around. And then suddenly this projection goes to the bud. And so this seemed like an ideal process to try to study at the molecular level to understand what's going on. And because it was yeast, we were able to do a screen for mutants that can't do this. And basically, we just did a tiny screen. I had just started my lab. We didn't have much funding. And we grabbed the first two pathways that we obtained. One of them totally makes sense. It's this motor-based transport by a myosin 5. And for those of you that were here last year, that is what I talked about. But the other pathway we got was this phosphoinositide signaling through Pi3 And what we imagine that this is doing is marking this part of the vacuole, because what you saw in that movie was that there was a lot of dancing around here and not down here. And when I get to the end of this talk, I'm speculating that maybe it was also part of forming these tubules, because I'm going to show you an event where this Pi3 is involved, that's involved in forming tubules. And so, phosphatidylinositol 3,5-bisphosphate is one of 7 phosphoinositide lipids that are found in plants and animals. And we think of how these phosphoinositide lipids work as through cascades. And there's not a lot of examples where we really have pinned down a cascade. And I'm going to be showing you a new one. But what we think of as a cascade is, so phosphatidylinositol, I like to think of as a structural lipid. It's part of what gives the membranes their properties. It's relatively high abundance. But Pi3P, which is made by phosphorylation at the 3 position, that is very low abundance. And then it serves as a signaling molecule by itself, and it can be converted by Fab1 or PIC5. So the Fab1's the yeast name, PIC5's the mammalian name, to Pi3P2. So what we imagine is that there is a membrane subdomain with phosphatidylinositol, and then there's a signal, and VPS34 or another Pi3 kinase is activated, phosphorylates the 3 position, and now you bring in these downstream Pi3P binding proteins that maybe don't always see each other or aren't always active, and now you've assembled a complex machine. And what I find is fascinating is that one of those Pi3P effectors is actually Fab1 PIC5, which has a 5 domain that binds to Pi3P. And now when it's activated, it's going to make Pi3P2. And then what's going to happen is the Pi3P binding effectors are going to go away, and the Pi3P2 effector proteins are going to come in. And so now we've had a cascade of coordinating event at a very special subdomain in space and time. Now, the other thing about Pi3P2 that really fascinates me is that we found a condition in yeast, hyperosmotic stress, where we could actually see dynamic changes in the lipid at the whole cell level. And so here are yeast happily growing in normal media, and then we add high salt in this case, or you could add high sorbitol, it's the hyperosmotic shock. We'll do that at 0 time. And then we do not perturb the cells for the rest of the experiment. And what happens is we see that in response to the signal, Pi3P2 levels skyrocket up. By 1 minute, they're already up 5-fold, and this is a 20-fold increase. And then even though we still haven't perturbed anything except for the 0 time point, there is a plateau and this precipitous drop. And so what it looked like to us is we're studying adaptation. And I think that is what we're studying in this case. And it's being regulated in some part by Pi3P2. And so because studying signaling in the yeast is considered kind of cute and not very important, we decided to make mice that we predicted would have half the normal levels of Pi3P2. And we learned a lot from these. And in collaboration with Miriam Meisler's lab, we found out that defects in this PIC5 pathway underlie several neurological diseases, neurodegenerative diseases. And in the case of a total FIC4 null mutant, it leads to this infantile death and affects multiple organs. And so this pathway is important in multiple organs, which makes sense. It's conserved from yeast. And somehow it's particularly important in the brain. And the other thing that we learned from these mice is that PIC5 is not only responsible for Pi3P2, but through myotubularin action on Pi3P2 is also responsible for almost all the cellular pools of Pi5P. And so now when we inhibit PIC5 or we do genetic manipulations, it's important to keep in mind that we're affecting both of these lipids. And right now, we don't have a good way to separate those two lipids, to separate the functions of those two lipids. Now, one other property of phosphoinositide lipids that I want to bring up is that when people first started working on them, the dogma in the field was that they marked organelle identity. And this has been a very hard concept to shake. I mean, it just sounds so easy and simple. And Pi3 was supposed to be important for lysosomes and autolysosomes. And then Pi4 was supposed to be important at the plasma membrane. But the more that people get into studying these phosphoinositide lipids, the more clear it is that they are actually working on multiple different organelles. And we imagine that they're just working in subdomains. And so because we saw those interesting neurological phenotypes in mice and because of the human diseases, we did some cell biology studies and electrophysiology studies in primary hippocampal neurons. And we found to our surprise that even though we thought PIC5 was going to mostly be working at lysosomes, it was important for the step in recycling AMPA receptors back to the, from internal storage back up to the postsynaptic membrane. And we also were not quite able to distinguish whether it was also responsible for the endocytosis. But what we saw was that when we stimulated the cells, there was a rapid change in AMPA receptors that required PIC5 activity. And so that suggested that PIC5 is acting elsewhere in the cell as well. Now, to get into this further, we started thinking about what would be a simpler cell type to study. And we were very attracted to papers from a few labs that reported that PIC5 is required for cell migration, although none of those papers explained how that was occurring, like where was PIC5 during cell migration. And so we found in our hands as well that using a scratch assay, we could see that if we acutely inhibited PIC5 with apilimod or this YM compound, that the wound that from a scratch assay was not healed in the same time frame. And so we wanted to ask the opposite. What if you had too much PIC5 activity? And we had developed a dominant hyperactive PIC5 mutant. And we found that when we expressed this mutant, we actually had the wound close faster. And because of this bidirectional activity, we became more convinced that maybe PIC5 was having a direct role. Don't forget, these assays are going over a relatively long period, 12 hours or 24 hours. And so we weren't quite sure if this was something that was way downstream of PIC5 or was proximal to PIC5 activity. And so we asked these questions: how does PIC5 play a role in cell migration? And where is the PIC5 pool that participates in this process? And so what I'm going to be telling you for the rest of this talk is that PIC5 functions in cell migration in part via positive regulation of the recycling of integrins. So as you remember from yesterday's talk by Ohad, integrins are really important in cell migration and in his cell spreading assay. And they're key to forming focal adhesions, and they have roles even prior to the focal adhesions in attachment of the cell to the extracellular matrix. And so now we had a much shorter assay we could look at in a 1 hour time point. When we looked at surface levels of beta-1 integrin in this case, we found that they were much lower compared to uninhibited cells. And then we wanted to look at the converse. What happens when you have hyperactive PIC5? And we found that in that case, more beta-1 integrin was driven to the cell surface. Now, those changes at the cell surface could be due to modulation of endocytosis or modulation of recycling, or both. And to look at endocytosis is much more difficult because as things that are endocytosed, they're recycled to the cell membrane, to the cell surface. But we can look at recycling by doing a pulse chase assay. And that's what I'm going to show you next. So what we did was a pulse where cells were incubated with a fluorescently labeled antibody for an hour. And then the cells were acid washed to strip off the antibodies that hadn't been internalized. And the cells were fixed to see what they looked like at the 0 time. And then we could do the chase on the unfixed cells. And some of the antibody will come back out to the cell surface and could actually continue to recycle. So when we looked, what we found was that in the untreated, the vehicle-treated cells, there was still plenty of a receptor that had come back to the cell surface after that stripping off of the antibodies. But in the case of the ipilimumab treatment, and now we're doing a 15-minute time point or a 30-minute time point, so even faster, we see that the integrin is trapped inside the cell. So it looks like there's some defect in recycling. And we can look at the converse, the same type of experiment. But in this case, at the end of the experiment, we again strip off the surface antibodies so we can just measure what's inside. And this gives us a more accurate measure. And what we can see is that in the wild type or the vehicle control, much less is trapped inside than in the ipilimumab treated cells. And so there are several ways that plasma membrane proteins can be recycled from these endosome compartments. And I'm showing you the ones that are the best studied and appear to be carrying most of the receptors. So the best characterized is this retromer complex. And this has been studied for several years. And it covers it returns several receptors to the plasma membrane. But integrin is carried by this other complex over here, which is called the retriever complex. And it's architecturally similar to the retromer complex, although most of the proteins are unique to the retriever complex, except for VPS29 is shared by both of them. And they both are regulating the wash complex. And so we looked and we found that endogenous, so using CRISPR-Cas9 editing, Guangming Lo in the lab made an HA tag on endogenous PIC5. Oh no, I'm so sorry. Okay, made a HA tag on endogenous PIC5 and found that it co-localizes with this retriever subunit, VPS35-like. And then we tested at least one subunit from all of these protein complexes. So I should have said at the beginning that the retriever complex is quite complex. There's the wash complex, which is organizing actin. There is something called the CCC complex. There are these retriever subunits. And there's the sorting nexons, NIC17. And so we took at least one subunit from each of these complexes, and PIC5 showed strong co-localization with all of them. And so what was known prior to our studies was that this complex of proteins assembles. The wash complex is involved with polymerizing actin, and then there's tubules that are formed and recycling back to the plasma membrane. And so we wanted to look at this further. And so we inhibited a PIC5 acutely for 30 minutes. And then we looked at all of those same subunits that I just showed you. And what we saw was striking. We saw that a member of the CCC complex, command1, was much lower, but SNCC17, the sorting nexon, was much higher. And we started to think about it more, and we realized that SNCC17 had already been shown to bind directly to Pi3P. FAM21 was also thought to bind to Pi3P. And what about over here, the CCC complex? And so we're thinking, well, maybe Pi3P is recruiting the SNCC17, and now we're inhibiting Pi3-5P2 levels. And I should show this here. So basically, when you inhibit PIK5, you see less Pi3-5P2 and less Pi5P. But what I failed to explain is you also get this huge increase in Pi3P. So we wanted to control that better. And we took a guess that maybe VPS34 was the Pi3 kinase that was contributing to this retriever pathway. And so we found conditions where we only partially inhibited VPS34. And at least at the whole cell level, we saw we could return Pi3P levels to normal levels when we had an inhibition of PIK5 at the same time that we had a partial inhibition of VPS34. And then marching through all these different combinations, we looked at several subunits to see how they were behaving. And just to summarize this is what we saw. So we saw that when we inhibited with apilimumab, SNCC17 levels went down. And so inhibiting with apilimumab gives you an increase in Pi3P and less Pi3-5P2 and Pi5P. And so we thought, okay, that means an apilimumab gives you this increase in SNCC17, but with inhibition with just VPS34 gave you a decrease. And so for that, we concluded that SNCC17 is recruited to endosomes by Pi3P. And then we looked at what happens with the CCC and retriever complex. And what we found is that with a treatment by apilimumab, the levels of the retriever went down. And inhibition with VPS34, which would lower Pi3-5P2, Pi3P, and the sum total of all of those three lipids also gave us less. And so from this, we concluded that the CCC and retriever complexes require Pi3-5P2 and or Pi5P to bind to endosomes. And then finally, we looked at the WASH complex. And we found that the WASH complex changed when we inhibited Pi3-5P2 or when we used enough BS34 inhibitor and apilimumab to bring the levels of Pi3P down. But with apilimumab treatment alone, there was no change. And so from this, we concluded that the WASH complex is recruited to endosomes by Pi3P or Pi3-5P2 and Pi5P. Any of those three phosphonosides should work. And so now we wanted to know, was anything in the CCC complex binding to Pi3P or Pi3-5P2 or Pi5P? And we followed up on some studies from Hela et al. That were published, where they solved a high resolution structure of the command 9 subunit, and then modeling the command 1 subunit, they modeled a structure. They showed biochemically that it bound to Pi3-5P2, and that these three residues were critical. Now I have to explain that in their paper, they substituted alanines, and when they looked in the cell, it didn't perturb the ability of command 1 to bind to endosomes. So we thought maybe the alanines in the cell were at least helping it to stay on the membrane, but they could still be affecting function. And so what we did was we actually made charge reversal mutants. And now with these charge reversal mutants, we saw that there's less command 1 on the VPS35 endosomes. But even more important, when we did a recycling assay, and we're now looking at the non-recycled pool of beta-1 integrin, we saw that there was a defect in this mutant, and it was failing to recycle integrins back to the plasma membrane. And so together, we think that this is now an ordered pathway, that integrins and Pi3P are recruiting the SNCC17, and the WASH complex also can get recruited. And PIK5, which I explained has this 5 domain, would also be recruited by Pi3P. And now the WASH complex will start polymerizing actin And the PIC5 will act to form Pi3-5P2, and Pi5P can also form there. And then the CCC and retriever complexes are recruited. We don't know yet whether there's any subunits in the retriever complex that bind Pi3-5P2 or Pi5P. And I suspect that there are more CC subunits that bind to Pi3-5P2. And then once those are recruited, then these tubules can form to recycle beta-1 integrin and other receptors back to the plasma membrane. And so I think the bigger picture of this story is that there are so many cell surface proteins whose cell surface expression is regulated. And I think a lot of regulation is going on in these recycling pathways, the retromer pathway and the retriever pathway. And very little is known about how these are regulated. But judging from our experience with studying the AMPA receptor, I think that these are highly regulated steps, and perhaps the phosphonosietide lipids are just one of the regulators that are involved in this happening. And so to summarize, I showed you that the retriever pathway is regulated via a phosphoinositide cascade of Pi3P to Pi3-5P2 and Pi5P, and that the CC subunit COMAM1 is one of the proteins that's a downstream effector of Pi3-5P2 or Pi5P, and that PIC5 has regulatory functions at endosomes that are engaged in recycling. And this is particularly interesting because back in the late 2000s, both Peter Cullen's lab and mine published complementary pieces of data that suggested that PIC5 is also regulating from that same endosome traffic back to the TGN. And so it'll be interesting to see, are there other endosomal steps that are being regulated by PIC5? Which also raises the question of, did we just mess up the endosomes? And what we're seeing isn't really a regulated process, but just a dysfunctional endosome. And we don't think that's true because we've looked at EGF receptor under acute PIC5 inhibition, and its plasma membrane levels are not changing. So I don't think everything is going haywire. And then finally, I'd like to say that the dynamic regulation of phosphoinositide lipids and their participation in cascades is likely common for the organization of many subcellular pathways. And I think one thing that's difficult is that there aren't known motifs for Pi3-5P2 binding proteins. So you're not going to find those downstream effectors in the bioinformatic approach. And the kinds of proteomic and genomic studies that people are doing are often missing the PIC5 pathway in particular, because because they're just the proteins that make the lipids are extremely low abundance. The lipids themselves are still not amenable to mass spec approaches. And so it's still going to be a while, I think, before we can take these broad approaches, which would be great, to look at where Pi3-5P2 and Pi5P are acting in the cell. And so this is a picture from before the pandemic. The people who were most involved in this process were Tsai and Guangming, and they really worked closely together. It was Guangming's idea to study cell migration in the first place, and then Tsai got so excited by the recycling assay that he did many of those experiments as well. And I'll stop here.
Thank you, Louise. We have time for some questions before live questions, and then we move to the chat if there are some questions. Vitash?
Hi, Lois. Very nice. So I think on one side, it looks like you and I are on the same page about cargo-specific pools of phosphoinositides.
Yes.
That's what I thought. So I have so what happens if you take mammalian cells, and because I think Bob Michel's lab also showed that if you hit them with salt or hyperosmotic conditions, that Pi3-5P2 levels go way up. So what happens to integrin recycling if you, rather than doing the genetic trick, you just change the osmolarity of the medium?
You know, that's a great question. And these, as you know, these experiments are super expensive. We're still struggling to figure out what are the best conditions to activate PIC5. And we don't see it as large an increase as he reported. We see something like a 60% increase, which is much more modest. So I'm not sure. We're still fooling around with different growth factors. I think what's going on is that the yeast can tolerate a much higher level of hyperosmotic shock than mammalian cells. I mean, they're set up for that biologically by growing in vineyards and getting rained on, and then it's drying out. They survive all that. Our bodies aren't quite flexible like that. We really need to be at a certain osmolarity. And I think that because of the different pools being different places, the stimuli are going to hit just a subset of pools.
Right. So I think that actually works in your favor, right? Because you have a 60% increase. And if you have such a tight regulation, you might expect one arm of the endocytic pathway to be really pretty sensitive to this, whereas the other one isn't. And in fact, under osmotic conditions, you might actually expect the recycling pathway to be a target for regulation of membrane movement, right? So just—
I see what you're saying. Yeah, I think that's an interesting idea.
Any other question? Maybe I have one more comment than a question, but actually when you modify genetically one enzyme of the PI pathway, most people actually look the target phosphoinositide if it's modified accordingly, but as all these PIs are intertwined, you may also modify other PIs along the pathway. And I think most people don't do that. What do you think about that? Maybe more acute ways to modify, at least in a timely fashion.
Yeah, so that's a really good point. So I didn't explain anything about how we measure the lipids or how we know we had a hyperactive mutant, or those, like how do we know what those inhibitors affect? So the way that we do these measurements, we actually get to see all 7 phosphoinositide lipids simultaneously. And so when I showed you this is what changed, I didn't explain, but all the other phosphoinositide lipids, Pi4-5P2, Pi4P, PIP3, these were not changing, or N3, Pi3-4. These were not changing under the manipulations that we did. I think the problem with our assay is that we're stuck with looking at cell lysates. And so we're always looking at the total cell level. And right now there still isn't a good biosensor for Pi3-5P2. And even though there are some people that use a biosensor for Pi5P, it's controversial what it's really recognizing. And so this whole axis is hard to study. And also there are good biosensors for Pi3P, but I don't think they're seeing all the Pi3P pools either, because I think it's some kind of coincidence detection that they can recognize the Pi3P, but there's something else they're recognizing too. And so they're recruiting to specific membranes. And so we really need better ways still to look at these phosphoinositide lipids. And I think that the mass spec approaches, the NMR approaches, these are all becoming much more sensitive. And eventually it will be possible to do these kinds of things.
Thank you. Any other questions? I think there are no questions in the chat box yet. So thank you, Lois.
Wait, Lauren Jackson has a question.
We have one.
Go ahead, Lauren.
Thank you, Lois. That was lovely. I was wondering if you have explored something similar for retromer. And I guess my question is, I thought your point about different PI head groups on different subdomains of the endosomes is so important. And, you know, is that another way that you partition cargoes maybe more efficiently, right, into a retromer tubule versus a retriever tubular structure, right, with maybe a different cascade? Or I just wondered what you thought about.
Or is it actually acting in both? We don't know, because what's really interesting is, so we in yeast, when you do co-localization, the yeast are so small, you see co-localization. And mammalian cells, co-localization, if you see a 40% overlap, you're good, right?
Okay.
But what's interesting is VPS 35 and VPS 35-like in our hands and in the literature, totally co-localized. This is at the level of light microscopy.
Oh, wow.
So they're at the very, very same organelle. And that's why I drew them as sharing that wash complex, because I think they're really like right on top of each other. But to really answer your question better, I'm so glad you asked, because if you ever get to a high enough resolution structure, can you see if Pi3P2 is binding in there? Or Pi5P? I wouldn't be surprised. I think that would be
VPS
amazing.
35? Or to one
Yeah,
of the
I know all of those, because you're looking at a complex, right? With cryo-EM?
Yeah.
Yeah, so I don't know what member it's going to be.
Right.
It could be any of those subunits. So what you would be looking for is you'd need to have something close to the atomic level. And then you'd be looking for those basic patches, right?
Interesting.
So if you see those basic patches, you have my cell phone number, call me.
Deal. That was a great talk. Thank you.
Okay, thank you, Lois. You can read all the congratulations in the chat box. So we move on to the next talk that is from Jae-Woong Kim. The title is CAP regulates the self-renewal of embryonic stem cells and cellular reprogramming. Please.
Hello, do you hear me?
Yes, and you see your slide.
Okay. Hello, this is Jae-Woong Kim from Busan National University in South Korea. Thank Lucio Coco for inviting me in this nice this nice symposium, and also I'd like to express I would like to appreciate my previous mentor, Song Ryu and Pang-gil Seo, for recommendation to recommend me to nice symposium. And today I'd like to present the role of CAP1 in the regulation of self-renewal of stem cell and somatic cell reprogramming. It is well known that embryo stem cell have a self-renewal potential and can be differentiate better cell types. And stem cell can differentiate better cell type. Also, the somatic cell can be reprogrammed to pluripotent cell by a cell reprogramming process. It is well known that somatic cell can be reprogrammed to induce pluripotent stem cell by overexpressing stemness factor such as OCT4, SOX2, KRAF, CMYC, and Lin28 and NANO. Today I'm going to talk about two topics. First one is role of CAP1 in the regulation of OCT4 protein stability. And the second one is role of CAP1 in the regulation of Lin28 and LAT7 signaling axis. OCT4 is a key signaling, key stemness factor involved in the regulation of stem cell, self-renewal, and pluripotency. And OCT4 interact with various protein complex, chromatin remodeling complex, and pluripotent specter, and signaling complex, and histone modification complex, and DNA methylation complex. However, the molecular identity of OCT4 binding proteins during the somatic cell reprogramming are still unknown. To understand the signaling networks involved in the somatic cell reprogramming, it is necessary to identify OCT4 binding protein in somatic cell. To identify OCT4 binding proteins, we overexpress the tandem streptavidin binding peptide tagged OCT4 in HEC293FT somatic cell. and then affinity purified the octo-4 protein with streptavidin agarose, and then analyzed the protein profile by using LC-mass analysis. This is the Western blotting analysis. This is a control tab, and this is octo-4 immune complex. We found that this is octo-4, and we found that a lot of the proteins could be co-flipped with octo-4. And by using LC mass spec analysis, we've identified various nuclear protein could be co-flipped with octo-4. Among the octo-4 binding protein, we found that pontin, leptin, and cab1 were identified as octo-4 binding proteins. Also, we identified various nuclear protein as an octo-4 binding protein. By Western blotting of the immune complex, we found that DNA PK, PAP1 and Cu70, these are nuclear protein also co-flipped. We found that cab1 and leptin, pontin are co-flipped with octo-4 by immune complex from HEC293 cell. To make a long story short, we summarized the role of the leptin and pontin as a octo-4 binding protein. And this pontin and leptin make a protein complex and then interact with the octo-4 and then stimulate the transcriptional activity of octo-4. Thereby, this protein complex play a key role in the self-renewal and reprogramming of somatic cell by stimulating octo-4 transcriptional activity in embryo stem cell and a reprogramming step. Another octo-4 binding protein we identified is cab1. Cab1 is a very long chain protein and have a multicellular domain and play a key role in the multifunctional role in the various cellular process. The function of the cab1 include DNA damage response and regulation of ubiquitination of p53 and epithelial mesenchymal transition and epigenic control of gene expression. So cab1 is a play a key role in the cellular response. This is the summary of the first topics of the presentation. So this is octo-4, and octo-4, this is each ubiquitin ligase, and this ubiquitin ligase ubiquitinate octo-4 at lysine 133 resid, and then the ubiquitination of octo-4 results in the proteolytic degradation of octo-4. However, the interaction with the cab1 interfere the interaction with each and octo-4, thereby prevent the ubiquitination and degradation and increase the stability of octo-4. By Western blotting of the co-mutation of cab1 and octo-4, we identified that cab1 could be co-flipped to octo-4 in embryo stem cell. And so next question is, what is the role of the cab1 in the regulation of the self-renewal capacity and pluripotency of embryo stem cell? This is the growth of the embryo stem cell colony and silencing of the cab1 using shRNA, resulting in the spontaneous differentiation of embryo stem cell and deformation of a colony of embryo stem cell. And after silencing, 2 days after silencing of cab1, the expression of the carbon reduced. And interestingly, simultaneously, OCT4 and SOX2 expression also goes down after silencing of carbon. And after 5 days or 4 days after carbon, the expression of bimentin, alpha-synuclein, and other differential marker expression also goes up. So this is the quantitation measurement, and after silencing of carbon, OCT4 protein, SOX2, and nano expression goes down. To clarify the role of carbon in the differentiation of stem cell, we made carbon doxycycline inducible carbon expressing embryonic stem cell line. After doxycycline treatment, the carbon expression increased. So during the culture, we removed the LIF to induce the differentiation of the stem cell, and doxycycline-induced carbon expression increase the colony number, colony size, and prevent the differentiation of the stem cell. So doxycycline increased the number of the undifferentiated stem cell population and prevent the spontaneous LIF-deprived differentiation. So from this result, we conclude that carbon play a key role in the regulation of self-renewal capacity and pluripotency of embryo stem cell. So what is the role of carbon in the regulation of OCT4 expression in embryo stem cell? Is the next question. To clarify the role of carbon in the expression of OCT4, we overexpressed carbon in embryo stem cell after by a doxycycline-induced manner. Doxycycline treatment increased the carbon expression after 2 days after doxycycline treatment, and then we found that OCT4 expression also simultaneously increased after doxycycline treatment. However, carbon expression, KRF4, another stem spectral expression, was now affected by doxycycline treatment. So carbon specifically increased the OCT4 expression without affecting the messenger RNA expression of OCT4. And we found that the carbon overexpression reduced the degradation of OCT4 after treatment of a cyclohexamide or protein synthesis inhibitor. So in this result conclude that carbon prevent protein degradation of OCT4. And we found that carbon overexpression decreased the ubiquitination of the OCT4. So carbon inhibit ubiquitination and protein mesosome degradation. And next question is, what is the ubiquitination site of OCT4, implicating the protein stability? So there are bioinformatics analysis. We identified several, speculated several ubiquitination site, potential ubiquitination site, and we made a point site-related mutant, and we found that the K133 arginine mutant resistant to the carbon silencing decrease of OCT4. Also, we found that each is a ubiquitin ligase. Overexpression of each decreased the OCT4 expression. Interestingly, the K133R mutant resistant to the each dependent ubiquitin degradation of OCT4. So we conclude that K133 residue of OCT4 is responsible for the Caban-dependent regulation of OCT4 protein stability. So what's the next question is, what's the role of Caban in somatic cell reprogramming, and what's the role of the mutant? We found that the silencing of Caban reduced the somatic cell reprogramming, usually 1% cell reprogram. However, silencing of Caban reduced the reprogramming efficiency. Interestingly, Caban overexpression greatly increased the reprogramming efficiency. Interestingly, in the absence of Caban, the overexpression of the K133I mutant increased the reprogramming efficiency. So this result, K133 mutant, which is resistant to ubiquitin protein degradation that stimulates reprogramming in the somatic cell. So this is the summary of the study. Each ubiquitinate and then proteasomal degradation. However, the Caban interact with OCT4 and then detach each from the OCT4, thereby stimulate protein stability of OCT4, and then increase the reprogramming efficiency. Second topic of the presentation is the Caban in the regulation of Lin28. Lin28 inhibits LAS7 expression, and then the Lin28 and LAS7 signaling axis play a role in the stem cell self-renewal and also in cancer cell. This is a summary of my presentation. Similar to the first story, TRIM71, another ubiquitin ligase, ubiquitinate Lin28, and then resulting in the proteasomal degradation. Caban interact with the N-terminal domain of Lin28, thereby inhibit the TRIM71 dependent ubiquitination, and then stimulate the protein stability of the Lin28. We identified Caban as a Lin28 binding protein as a mass analysis, and we confirmed the protein-protein interaction by Western blotting, co-immunomutation of Lin28 from embryonic stem cell. And silencing of Caban reduced the expression of Lin28 and Lin28B. In contrast, overexpression of Caban increased the Lin28, Lin28B expression. And Caban overexpression increased the protein stability of Lin28 A and B subtype without affecting mRNA expression. And then we found that the Caban overexpression, Caban silencing increased the ubiquitination of the Lin28, Lin28B, and doxycycline-inducible Caban expression decreased the LAS7 expression level, mature expression level, and embryonic stem cell. So this is the summary of the presentation. In TRIM71, ubiquitinate in the cytosol, ubiquitinate Lin28, and then resulting in the proteasomal degradation of Lin28 isoform. However, in embryonic stem cell, Caban increased the Caban interfere TRIM71 dependent ubiquitination, and then increased the protein stability of the Lin28, and thereby play a key role in the stemness in embryo stem cell. This is the final summary of my presentation. Caban is a master regulator for protein stability of the reproductive factors. We've found that Caban interacts with OCT4 and SOX2 and NANOG and N28, and then increase the protein stability, thereby play a key role in the regulation of stemness in embryonic stem cell and cancer. This work was mostly done by my student, Eun-kyung Do and Hae-ji Moon, and I highly appreciate my previous mentor, Pan-gil Seo and Song-ho Ryu. Thank you for your attention.
Thank you, Jae-Ho. We have time for some questions.
Yes, I have one. Very nice study. Are CAP1 levels regulated by LIF?
Excuse me? Caban regulate?
So are CAP1 levels in stem cells regulated by LIF? So if you take LIF away, CAP1 levels drop. If you add LIF, do they increase?
Yeah. We found that the expression level of CAP1 is highly expressed in the embryo stem cell. And then during the differentiation, the expression of CAP1 goes down. So CAP1 expression is correlated with the self-renewal activity of the stem cell. But however, we haven't checked the effect of LIF on the expression of CAP1. That's a good question.
Any other questions? Maybe can you tell me, I didn't get this, how CAP1 is affecting ubiquitination?
So yeah, good questions, and thank you. CAP1 is a ubiquitinol ligase, and it has been regulated to ubiquitinate P53 and other protein. And so in my presentation, interestingly, it has an opposite role in the regulation of ubiquitination. It interferes ubiquitination of the OCT4 and N28 by other specific ubiquitin ligase. So in this my talk, CAP1 play a role in the negative role in the ubiquitinations of the OCT4 and N28 by interfering the ubiquitination process.
Thank you.
Can you understand?
Yes. Any further questions?
Yeah, can I ask a
Sure,
question?
sure.
You mentioned the link between programming and cancer, particularly EMT, which I think is very intriguing. So my question is, are EMT transcription factors, Twist, SLUG, and others, are they directly stabilized by these factors?
I know your mic is very hard to understand. It's cracking.
I'm sorry.
Yeah.
I'm not sure if I can do
I
anything about it.
understand,
The question was EMT transcription factors, are they stabilized by these factors?
Yeah, good questions. And we're also interested in the role of the CAP1 in cancer and EMT. And we also studied the cancer stem cell. And we found that the silencing of CAP1 reduced some of the stemness marker expression and some EMT marker in cancer cell. Cancer stem cell. So we are studying the role of the CAP1 in the tumorigenesis or EMT process but still, that story is still ongoing. So maybe next time I'm going to present.
Thank you.
Thank you. Any other question? So, well, if there are no further questions, I really want to thank the two speakers, Lois and Jay-Ho, for their great talks and for being sharp on time as well. And so there is a 5-minute break and we'll be back.
Thank you.
I called the guy. So you're the second one, Nico? Oh, yes. Okay, I think we can start now with the second session today. And the first speaker is Aurelia Lewis from Bergen, Norway, and she's going to talk about nuclear phosphoinositide 3 kinase signaling. Please, Aurelia, it's up to you.
Yes, thank you very much. Do you hear me?
Yes, yes.
Yeah. Okay, so yes, let me just go on screen mode if it wants to do it. Yes. So is it gone the other way now? Or do you see my main screen?
It's perfect.
It's perfect. Okay, great, because I see the opposite. Thank you. Right, so thank you very much for the invitation. And as Lucio started the symposium by introducing the nuclear aspects of PLC beta, I'd like to take you back to the nucleus a bit and show you how we think Pi3 kinase is signaling in the nucleolus. And as you know, Pi3 kinase are very well understood in terms of how they signal and how they activated and what sort of processes they activate from when they orchestrate these aspects from the cytoplasm and especially from the plasma membrane. However, many studies have showed that components of the pathway have been detected and found in the nucleus. And that includes also phosphoinositides. And just to recap, these are the phosphoinositides that have been detected via various methods, and that dates back from the time when Lucio was looking into this back in the UK. And in terms of the enzymes that metabolize these phosphoinositides, in terms of the kinase and the phosphatase, also several isoforms have been shown also in the nucleus, in various parts of the nucleus. And I would like to start by pointing you and focusing on the members of the Pi3 kinase. And in terms of the class 1 Pi3 kinase, there are several isoforms, as you can see here for the catalytic subunit, alpha, beta, and delta for class 1A. And one of them, P10 beta, which is ubiquitously expressed, is found in the nucleus, as well as P10 and reversing the reaction. Yes, and in terms, so we were quite interested in P10 beta in the cancer of the endometrial of the uterus. And that's because many, at the time, many cancers that were PTEN deficient were reliant on P10 beta activity. And in endometrial cancer, P10 is one of the genes that is the most deleted or lost. So we were quite interested in looking into P10 beta in this cancer. And we got a panel of cell lines here, of endometrial cancer cell lines, and then a reference cell line, which is immortalized but non-tumorigenic. And we did some fractionation. We isolated the nuclei and looked at the expression and the levels of alpha versus beta, and found that indeed beta was both cytoplasmic and nuclear, but its levels were increased in the nucleus in some of these cancer cells. Now, in terms of tissue, we did some immunohistochemistry, and we showed that in overall in the low tumor grade, P10 beta was confined to the cytoplasm in these nice looking glands, and in higher grades of this cancer, it was more restricted to the nucleus. And then the quantification of that, when we look at different tumor grade and looked at the ratio between nuclear staining versus cytoplasmic staining, there was an increase in nuclear detection in the higher grade, so a small decrease there. So we were interested in finding out, actually, to look a little bit more at the activity of P10 beta. And first of all, to look at the localization of P10 beta, and found actually that it was not only cytoplasmic, but also nucleolar in some cancer cells. So this is our nucleolar marker, nucleophosmin, and you can see that there's a nice colocalization in this cancer cell line. And in terms of the lipid, PIP3, we also found, so using high detergent methods, we also found that PIP3 was colocalized with a nucleolar marker within the nucleus. So, and we also showed that the presence of PIP3 in the nucleolus was due, at least partially due to P10 beta activity. And we got some MEF wild type and kinase inactive from Julie Guillermet-Guibert from Toulouse. And using these cells, we fixed them and probed them or incubated them with and a recombinant protein that we made, which is a GFP tagged pH domain of GRIP1, which recognize and bind to and can detect PIP3. And then we did some staining with the nucleolar marker. And as you can see, we find in the pH domain highlighting nucleolar structures, as well as cytoplasmic structures, whereas the kinase dead cells showed a decrease in the detection. Now, in terms of, we also used another assay, and again, we made our GFP recombinant pH GRIP1 recombinant protein and made a mutant that was known at the time not to bind to PIP3. So we probed these two recombinant proteins on lipid overlay assay, and shown these are spotted with different phosphoinositides and other phospholipids, and find that indeed the wild type interacts with the PIP3, but that the mutant didn't, but there's still some binding to phosphatidic acid. And then we use these two recombinant proteins in Hela cells and try to look at the co-localization of the detection of PIP3 signal in the nucleolus. So inside the, this is a nucleophosmin a marker for the nucleolar detection, and showed here with the quantification, as you can see, that the mutant indeed didn't recognize these signals from the nucleolus. Now, so as you know, the nucleolus is a ribosome factory, and many processes are important in the nucleus to achieve ribosome biogenesis, and it involves the transcription of the ribosomal RNA, cleavage, processing, various modification, and assembly with ribosomal proteins to make the small and large subunits. So these subunits then are exported to the cytoplasm, ready for protein synthesis. So we focused on one aspect so far, and that was the transcription here. And we looked at the levels of 47S in our panel of endometrial cancer cells here compared to our reference cell line, and we see that there is an increase in the levels of the transcript. And then we chose a couple of these cell lines and treated them with a selective P-anten beta inhibitor, and so a decrease in the levels of the transcript. And this was correlated with a decrease in cell division using another P-anten beta inhibitor. Here, as you can see in the light gray, the cells plateau and don't divide anymore after some days of treatment. So overall, what we're thinking at this stage, and still plenty to do, is that in how P-anten beta contributes in cancer, it might cooperate with the cytoplasmic role of the Pi3 kinase pathway with nuclear activity, and by at least increasing the levels of the transcript. But what we were wondering is how PIP3 is maybe contributing to P-anten beta downstream effects. So, and as you know, those PIPs are involved in binding to proteins and can affect their function through binding. So we developed a method to map the interactome of phosphoinositides from the nuclei. And this is by using SILAC labeling and isolation, so nuclear fractionation. And then once we have nuclei, we incubate them with a high dose of neomycin, which is a polybasic antibiotic, so positively charged, which it competes with between protein and phosphoinositide interaction. Of course, we can't exclude any other charge interaction due to DNA or RNA. But anyhow, we collect our proteins that are extracted from this treatment and do a pull down with PIP3 or control beads and identify proteins and do some quantification. So we identified a number of proteins in complex with PIP3. And when we search for interaction domains, so the canonical domains, we hardly find any proteins, but in the nucleus, it's less the case. But we find many proteins that have polybasic motifs. And in terms of the function, the processes that these proteins are involved, they're involved in many different processes, nuclear processes, and these are the ones that are enriched compared to nuclear or a whole list of nuclear proteins. And some of these processes are also even more enriched when we only look at the proteins that have at least one of these polybasic motifs. Yes, so then we've decided and the important thing from this message is that when we compare our list of proteins to a nucleolar database, so knowing that PIP3, by then we knew PIP3 was localized not only in the nucleoplasm, but also in the nucleolus, we actually found that depending on the database that we look at, between 29 and 40% of them belong to are detected in the nucleolus, so have nucleolar roles. So we try to focus on a protein to validate some of our findings, and we focused on this particular process, DNA repair, and one of the proteins that were bound to PIP3 or in complex with PIP3 was polyadipose ADP ribose polymerase 1, PARP1. And it is a well-known nuclear protein. We saw that in the previous talk in one of the slides, but it was also found to be nucleolar. So it has roles in the nucleolus, but these are less understood and well characterized. So first we decided to validate that this protein binds to phosphoinositides directly. So we expressed a recombinant protein, a GST-bound tagged protein. I found with our overlay assay that it indeed could bind to many different phosphoinositides. And then we also did a lipid pull down using the same lipid pull down that we did for the mass spec, but this time looking at in a Western blot, and we found indeed that GST PARP1 interacted with the PIP3, but it also interacted with 3,4-P2. And in terms of when we looked at the staining, we could see that a number of cells localize PARP1 in the nucleolus here, together with PIP3, but not all cells. And in terms of 4,5-P2 or 3,4-P2, which are two phosphoinositides that have been detected previously in the nucleus, but there is a little colocalization with 4,5-P2. However, with 3,4, we saw some cells co-localizing in the nucleus speckles with the PARP1. So there seems to be different pools of PARP1. These are growing cells, asynchronously growing cells. And there is a pool in the nucleolus that seems to co-localize with PIP3 or or with 3,4-P2 in the nucleoplasm. So we wanted to find out the interaction site within PARP1, and this protein has many domains, a DNA binding domain with different zinc fingers, catalytic domain, and an auto-modification domain. So what we did is we split this protein in different fragments here to make it a little bit systematic. And again, we produce these different fragments, GST tagged, and we found that out of these, I can't remember how many there were, 6 fragments, 3 of them showed some interactions with phosphoinositides with our overlay assay. So the DNA binding domain and this linker region here between these two domains. So again, this protein didn't have any canonical phosphoinositide binding domain, but it had different motifs when we looked at these 3 different regions here. And so these polybasic regions, we've mutated them to either mutated them or we deleted a whole chunk of these regions here. And again, we've tested them in a lipid overlay assay and found that in particular the 2 regions that we deleted in the zinc finger 3 here were decreased quite a lot, the interaction. And this was the same for our linker. We also did NMR with the linker because this was the shortest, the smallest piece of the protein that we could work with in NMR, and I'm sure we could do better, but that's all we could do. And this also helped us to identify these lysine residues in HSQC assay. So again, yeah, these lysines were important for the interaction. So far we've just identified the interaction, but what we really want to know is how interaction can affect the activity of PARP1. And I can see that I would anticipate that you would want me to tell you about it, but these are still preliminary results. But what we have done so far is we've tried to look at PARP activity in MEF cells, wild type, and the P10 beta kinase inactive cells. And we can induce PARP activity, and we can detect it. We can detect the modification, so this PARylation, these polymers of poly-ADP ribose using an antibody, and we can detect it on the stimulation of hydrogen peroxide. So we can detect this PARylation. It's very difficult, so we try to use methods that would look at nucleolar detection of this modification, but we haven't managed to really recapitulate what the previous reports had shown. But we see some PARylation together with this fibrillary is another nucleolar protein. So we see some of it. But so far, we have not been able to detect much changes. This is just one experiment. So these are very recent data from a master student. So we can still see detect PARylation within the nucleolar pool. So I think we need to use different DNA damage agent to maybe see a difference there, or maybe not. So we're still working on trying to figure out how PIP3 binding can affect the function of PARP1 and other proteins that we've also identified. But so far we've actually found that the Pi3 kinase P10 beta is elevated in some cancer. The nucleolar activity is increased at least at the transcription level, and that's due to to the activity of the enzyme. We have looked into PIP3 interactors from the nucleus, and many of them are nucleolar proteins, and some don't necessarily have bona fide nucleolar activity, but additional nuclear activity connected to the nucleolus. And then here I've showed that we focused on one of these proteins, PARP1, but we still have plenty of things to do. So with that, I would like to really highlight the work of the students, that PhD students, but also many master students, actually, that have contributed to this work. So many master students have had the pleasure of working with, and also the collaborators, Clive De Santos, he's done the proteomic work on the PIP3 interactome, Julie for the cells and continual chat over P10 beta, and a collaborator in Bergen for the tissue immunohistochemistry. And with that, I would be very happy to take questions. And
we thank you.
Thank you.
Thank you for your talk. Now it's open to questions. It's Nicholas.
I have a question.
Great presentation, Aurelia.
Thank you.
First, I have a comment. I think the PIP assays are not very reliable regarding the specificity. They mainly tell you that it's an ionic interaction. I think liposome-based are probably more suitable. But my question is actually, as you see the enzyme and the product, do you know if you have also Pi4-5 PIP2 in the nucleol? And the second question is, did you identify in your proteins phosphatase that may recycle back PIP3 to PIP2?
Yes, so 4,5P2 is, when you look at the nucleus, is essentially in the nucleus speckles, but there is a minor pool also in the nucleolus. So you can detect amino polar 4,5p2. So, but so it is there. And in terms of reversing the reaction, P10 has been shown to be nuclear, and there was a report showing its nucleolar also localization, and also a splicing form of P10, which is also nucleolar. So there was a report in NatureCom a couple of years ago, or 3 years ago. We've tried to get construct from them, but we've never heard. We wanted to use their construct to really target P10 to the nucleolus, but yeah, they never responded. So it would be nice to be able to target the phosphatase or SHIP2. Maybe we could target SHIP2 to the nucleolus, and that would be a way to manipulate PIP3 levels.
Christophe Renaud has a question. Christophe, why don't you go?
Yes, I wanted to ask you, how do you see nuclear PIP3 to be regulated? In the same way or in different ways compared to plasma membrane PIP3? And this relates, of course, to the P10 or SHIP2 expression in the nucleus. And the second question, H2O2 is one of the best ways to increase Pi3 for P2 and to also increase PIP3 at the plasma membrane. So does H2O2 also affect the lipids in the nucleus?
So what was the first question? Maybe I go to the second question now. But they're connected. The regulation, I guess, of how do we make PIP3 there in the nucleus or in the sub. Well, I can't tell you, but I would imagine that the mechanisms are quite different to the membrane. So in terms of the regulatory subunit, P85, which is very much involved in regulating the activity, targeting the catalytic subunit at the membrane. So P85 is also nuclear, but how do they work in complex? We haven't even started to look into it. And how do they actually utilize the substrate? In what form the substrate is present in the nucleolus or anywhere in the nucleus, this is not very clear. Apart from studies from Ray Blind, who showed that PIP2 and PIP3, the acyl chains are shielded into a ligand binding pocket, so thereby exposing the head group for modification. So that's a possibility, but then not these nuclear receptors are, yes, it's an explanation, but it doesn't explain everything as to how the substrate is presented to these enzymes in a non-membrane area. But what's interesting, and there was a comment, if I may say that, there was a comment 2 days ago about these RNA binding proteins in virus, and how they the binding site compete for membrane binding and for RNA, and then these phosphoinositide are found actually in areas of the nucleus where it's packed with RNA. So there's also some charge competition. So how are they actually found there? Whether they're in sort of mice cells, it's not very clear. But I could go on. Sorry, I don't want to use so much time. I'm not sure if I answered everything you asked.
Lois, you have a question?
So
very
Lois.
nice talk. I'm sorry. So you found this by looking at cancer cells, and have you thought about what cell types might actually just do this naturally? Would it be rapidly proliferating cells? I'm just trying to think about whether there's another cell type you could look at, and that might give more depth to the project.
Yes, that would be really interesting to do that. Yes. So we've looked at cancer cells, but we've also tried to manipulate them in terms of the cell cycle. So that's the only way we could sort of not see PIP3 in the nucleus, but of course after mitosis, it takes some time, quite some time, before p-10 beta goes back in the nucleolus and PIP3 is made. So that's the only model we've got so far to try to show that it's not anywhere, or at least just cancer cells as such. So highly proliferating condition. But I think it would be really good, would be really neat to show that in other systems.
So I have a very quick question that I hope, and a quick answer. So there's two ways of thinking about this. One is that one generates a PIP3 pool and then one recruits, let's say PARP, for example, to the site. The other possibility is PARP is a binding partner for 110 beta, right? And essentially you have a complex and then you have local PIP3 synthesis that regulates this. From your data, can you comment on which model you would like better at this point?
Second one would be really cool. So a complex with because you have and as we know, PIPs are really localized. They have a very local action, so that would be really neat if it was, you know, complex, and then it's a little bit like what Ray is showing, because that's what it's a different system, of course. So that's not a short answer, sorry, but okay, number 2, I would say, would be really neat.
Okay, it's a very last question.
One last question. How, do you think P10 shuttles back and forth from nucleus to the cytoplasm? Because as a cancer biologist, we know that the P10 in nucleus is pretty dangerous. It's pretty tumorigenic, and we want it to stay in cytoplasm.
Yes. I don't know. We haven't looked at that in terms of the shuttling. But and then we've especially studied cancer cells that are P10 negative. But no, I don't know. I can't answer your question.
Okay, thank you again, Aurelia. And now is time for Nicholas Vitale from Strasbourg. He's going to talk tonight about mono and polyunsaturated phosphatidic acid regulate distinct steps of regulated exocytosis in neuroendocrine cells. Please.
Okay, thank you, Lucio. I want to thank the organizers for such a great meeting and taking such great care of the participant locally. So I will present you our data showing that a specific lipid and specific members of this family of lipids are controlling regulated exocytosis. So regulated exocytosis is the process that allows in our organisms specific cells to secrete molecules such as a neurotransmitter, neuropeptides, but also hormones. And in our lab, we are using chromaffin cells as a model. Those cells are present in the adrenal medulla gland. And as you can see here, they are highly specialized cells that contain more than 20,000 secretory granules packed with the stress hormone catecholamines. And upon a stressful situation, those cells secrete catecholamines in order to allow the organisms to adapt to this stressful situation. This process—
Hey guys, sorry about this. I don't think any of us that are on Zoom can actually see the screen. So there's a few comments in the chat. I think they were united on that front.
All right.
So maybe just share the screen.
I'm not seeing the slides.
Those cells— All right, I'll just go back to the previous slide that shows the model. that those cells are highly specialized and secrete catecholamines.
There's a few comments in the chat. I think they're united on that front.
Yeah, it just came up.
So maybe just share the screen.
It's okay for everybody? Okay.
It's working. Thank you.
All right, thank you. So this process is a multi-step process that starts by the biogenesis of the vesicles at the Golgi apparatus. These vesicles are then transported to the cell periphery, and then they undergo successive steps. And you already seen this cycle yesterday in Dan's presentation. The vesicles are first docked at the plasma membrane, following a priming step, and then they fuse in order to release their content into the extracellular medium before being re-endocytosed in order to maintain cellular homeostasis. And these endocytosis vesicles can be recycled back to make new secretory vesicles. As you are probably aware, many proteins in this process are well known, including the snare proteins, but also the cytoskeleton that play a crucial role. But actually the function of lipid into this process is much less understood. And this is rather intriguing, since actually lipids are the main constituents of these vesicles. So today I will present you the role of one specific lipid, which is phosphatidic acid. This tiny phospholipid is actually a very interesting lipid. It has first structural function, as it is the substrate that generates most phospholipids in membrane, but it has also signaling function, as it can be synthesized by many enzymes, including phospholipase D, acetyltransferase, and deacetylglycerol kinase, and can be also converted back to the original lipids in order to modify the level of this lipid in one specific location and one specific time. What makes phosphatidic rather interesting is that it has a negative charge, so it can interact with positive domains in protein, but it also has, through its small polar head group, a cone-shaped structure, and these cone-shaped structure are supposed to be important for membrane trafficking, especially which require membrane deformation. And this is especially the case when actually phosphatidic acid is generated by members of the phospholipase D enzyme family, as it converts phosphatidylcholine, which is a cylindrical lipid, into phosphatidic acid, which has a conical shape. So at one specific location, you are generating locally a membrane curvature that would favor membrane deformation. So in order to probe the function of these PLD enzymes, we generated PLD knockout mice, the two main isoforms, PLD1 and PLD2, and measured the catecholamine level into the blood of stressed mice. So today I will just present you one condition, which is actually right after natural delivery. There is a sharp rise of catecholamine that can be measured after 1 hour, and it rapidly declines to basal level within 24 hours. And you can see from this scheme that PLD knockout mice show a dramatic reduction into the amount of adrenaline and noradrenaline released into these newborn mice, indicating that the endogenous PLD1 contributes to the release of these catecholamines into the blood. So what is actually the function of this PLD into the release? Either it can modify the level of release or the secretory granule content of these catecholamines. So first, we did electron microscopy analysis of the PLD1 knockout chromaffin cells, and we reported a small but significant reduction into the number of secretory vesicles. But this small reduction is probably unlikely to contribute to explain the rather strong inhibition into the amount of catecholamine that we found into the blood. So we further analyzed our mouse, and we actually did not see any difference or significant difference into the adrenal gland level. But there is also a small modification to the diameter of these vesicles that are a little bit smaller. However, altogether, this probably not recapitulates the strong inhibition of catecholamine that we detected into the newborn mice. So we actually estimated the release capacity of those cells. And this slide summarizes what I have presented last year to you. Using an electrochemical approach, which is called carbon fiber amperometry, from which we can actually record single exocytotic event from single chromaffin cells. associated with electron microscopy of rooftop membrane sheet that we prepared from chromaffin cells. And this using, in association, genetic, silencing, and pharmacological approach to modulate PA levels, we were actually able to show that monounsaturated forms of phosphatidic acid regulate secretory vesicle docking, and polyunsaturated forms of phosphatidic acid regulate the late fusion step of the process. However, in order to continue this work, we tried to determine whether this PA also contributed to the other steps of this process, and this is what I will describe to you today. So the first approach was to monitor secretory vesicle dynamic in cells, and to do that, we did a 3D movement analysis of NPY RFP expressing cells, chromaffin cells. You can see in the movie that the granules are quite moving into the cell. To make a long story short, these granules display three types of movement, either a free movement, and those granules are mainly found into the center of the cells, probably right after the exit of the Golgi. Then a population of the granule have a cage movement in a very narrow area. Those probably correspond to the docked granule at the plasma membrane, since those are highly enriched at the cell periphery. And finally, there is a third population that has a directed movement, and those probably correspond to the granules that are transported along the microtubule into the cells. And what we found is that after application of a PLD inhibitor, FIPI, there is a significant reduction into the velocity of these granules, indicating that indeed PLD-generated phosphatidic acid may also contribute to the dynamic of these secretory vesicles. Next, we investigated whether PA synthesis by PLD1 is involved in compensatory endocytosis, and to do that, we used a dopamine beta-hydroxylase internalization assay. DBH is an enzyme that is present exclusively into the inner leaflet of the secretory vesicles and becomes accessible to the medium only upon fusion of the secretory granule with the plasma membrane. And when we put an antibody that specifically recognizes this antigen, it can be captured and follow the retrieval of these secretory vesicles. And by doing an image analysis by Euclidean distant matrix, we can determine whether those those endocytotic vesicles are internalized and at distance from the plasma membrane. And on the image, you can see we measured endocytosis after a short time or a longer time, 5 and 30 minutes. And what we found is that after 5 minutes, the PLD inhibition reduced the internalization, which is not anymore the case after 30 minutes, indicating that PLD probably regulates the early step of the endocytotic process, but maybe not the later steps. So to summarize this part, I've shown you that mono and polyunsaturated PA produced by PLD1 regulate different steps: the granule biogenesis, their transport, their docking, their fusion, and their retrieval back to the endocytotic compartments. But where is actually phosphatidic acid synthesized in cells? And to address this, we used a specific sensor that we developed in the lab for phosphatidic acid. And when this sensor is used, you can see that it is highly found at the cell periphery, at the plasma membrane, or the secretory vesicles that are docked at the plasma membrane upon cell stimulation. And when we did a morphometric analysis, we found that there is an enrichment, that there is a threefold enrichment of this PA sensor when a secretory granule is directly facing the plasma membrane, indicating that there is a specific enrichment of this PA at the site where the granules are docked. So we continue this work and use our membrane sheet preparation, and you can see here, you can appreciate the difference into the actin network that is surrounding the secretory granule when you compare the resting and the stimulated condition. You can clearly see that the actin network is restructured and actually forms large bundles that anchor the secretory vesicles to the plasma membrane. And we believe that in addition to anchoring, this structure probably also provides some force to push and to release all the content of the secretory granules. And you can see on this slide that the PA sensor is highly enriched close to these actin bundles. And from time to time on secretory vesicles that are starting to get empty, so become less dark, we can also see that the PA sensor is found below the secretory vesicles, probably at the fusion site itself. So we have shown now that phosphatidic acid is synthesized at the plasma membrane, but what is the type of phosphatidic acid? As you all know, every phospholipid comes in different flavor based on the fatty acid chain. So actually we performed a detailed mass spectrometry and mass spec analysis of phosphatidic acid species from purified plasma membranes from resting and stimulated condition. And you can see here that upon cell stimulation, there is more than 10 different forms of phosphatidic acid species that are highly enriched upon cell stimulation, and some of them up to 10-fold enriched. And you can appreciate also that among those phosphatidic acid, there is mainly mono and polyunsaturated forms, which is in line with the finding that I've shown you earlier, that those are the forms that regulate the docking and the fusion step. So to summarize, these two types of phosphatidic acid are produced at the plasma membrane near exocytotic sites during exocytosis. So the next question we wanted to ask is actually, what is phosphatidic acid doing into this process? And one of the first arguments is that it may change membrane topology, as I've already mentioned. And this is based by basically largely on biophysical experiments that suggest that a cone-shaped lipid, such as this one in green, are favorable of this membrane curvature needed for the hemifusion and the fusion steps. So we thought that actually providing a lipid that has the opposite geometry into the outer leaflet of the membrane and of the plasma membrane, such as a lysophosphatidic of a lysolipid, would actually recapitulate this function of phosphatidic acid. So what we did, we applied lysophosphatidylcholine to secretory cells in condition where it does not affect secretion on its own, and on this condition, it can partially restore the inhibition that we see with a PLD1 silenced cells, indicating that at least part of the function of the PA produced by PLD1 is indeed linked to a curvature effect. But this is probably not the only thing it's doing. So we also went to determine whether some protein targets could explain some of the function of phosphatidic acid. And the first target we identified is called chromogranin A. This protein is a well-known constituent of the inner of the secretory vesicles. It is actually known to play a key role into the granulogenesis of these vesicles, but the exact function is not clear. We identified within the carboxy terminal part of this protein a region that has all the features to interact with phosphatidic acid. It is a polybasic region with alpha helical amphipathic structure, and in vitro on liposomes, it is able to bind to liposome containing PC and PA, but not PC alone. In order to see if this has an effect on the biogenesis of the vesicles, we undergo three different types of experiments. The first one is to apply CGA on fluorescence cube, and you can see that the application of CGA induces the generation of the budding of vacuoles, of vesicles, indicating that indeed when PA and CGA are together, there is a change of membrane topology. Then we performed atomic force microscopy on supported by layer containing PA, and the provision of CGA also changes the topology of the membrane. As you can see here, there is clunches of membrane that appear when chromogranin A is added. So finally, to more directly prove a direct role into the biogenesis, we use a cell line, COS7 cells, that normally do not have secretory granule. However, when they express chromogranin A, they do form secretory granules. So we expressed the wild type chromogranin A or a chromogranin A deleted of this PA binding domain. And under those circumstances, we see a dramatic reduction into the number of secretory granules formed, indicating that it did part of the function of PA here is to recruit chromogranin A that will deform the Golgi membrane and will generate the granules. And then the last potential interactor of phosphatidic acid is syntaxin 1A, one of the snare protein. We also identified a potential PA binding motif, which is in a juxtamembrane domain between the transmembrane domain and the snare motif. When we mutated these basic residues to alanine, we completely lose the ability to bind to phosphatidic acid. And in chromaffin cells, the secretory activity is modified with a reduction into the number of events and modification of individual spike parameters, very similar to what we see into the PLD1 knockout mice, indicating that maybe syntaxin 1 is indeed one of the interactor of phosphatidic acid into these docking and fusion steps. And finally, we also find that phosphatidic acid, either the mono or the polyunsaturated forms, interact very well with this syntaxin PA binding domain, with almost no interaction with saturated PA and much less with PIP2. So to summarize our finding, we have shown that PA is involved into the biogenesis of the vesicles, their transport, their docking, their fusion, their recapture by compensatory endocytosis. We have identified for 2 of those steps the type of phosphatidic acid that are involved. This is rather interesting since polyunsaturated lipids are known for a long time to be very important to prevent cognitive decline. So it may be that this type of phosphatidic acid form may actually contribute to a neurotransmitter release. But this remains to be actually fully evaluated. But we believe that the tools that we actually have in hand are not adequate enough in order to study this process, for example, in neurons that are very rapid systems, but also they do not allow to precisely determine the regulation of phosphatidic acid at a very precise time frame, determining the enzyme involved in the genesis, but also in the hydrolysis. So the whole metabolism needs to be fully appreciated. So in order to do that, we are currently developing a comprehensive tool to study phosphatidic acid. So we have generated a targetable set of enzymes that we can direct to specific membrane compartment upon light illumination. So upon few milliseconds, you can recruit any of those enzymes that are involved either into the synthesis or the degradation of phosphatidic acid to any specific compartment in cells. We also developed new photoactivable PA sensor. I don't have time to go too long into that, but the actual sensor have bias since they actually quench the natural phosphatidic acid during their expression. And this is the case for all type of sensors. So we want to develop sensor that can be activated again upon light stimulation. And finally, we are synthesizing synthetic phosphatidic acid that can be caged and uncaged upon will. So you can produce locally through a flash of light phosphatidic acid, depending on the species in one specific location. So finally, I want to thank all my collaborators that are involved into this project, especially Emeline Tanguy and Alexandre Wolf that is now continuing her work. I also want to thank our collaborators, our fundings, and I want to thank you for your attention, and I'm happy to address any question.
Thank you. Very nice talk. Questions? First of all, okay, on you go. Then there is...
Is it working? This is an excellent talk. So do you think the same way it's working post-synaptic vesicle in neurotransmission, and also when the presynaptic, is it forming the vesicle back?
Yeah, in neurons you have synaptic vesicles, but you also have secretory granules. So the process is maybe a little bit different between the two types of vesicles. For instance, the speed is much faster for synaptic vesicles, but we have shown almost 20 years ago actually that in aplesia neurons, when you compete with PLD, you also affect secretion, indicating that probably it's also involved in neurosecretion in neurons. But there is not much work. The problem with neurons is that they are not so easy to work, at least compared to our cells. But as you have seen from Dan yesterday, the production of phosphatidic acid during the endocytosis might be actually involve another set of enzymes, but in our cells, the effect we see is rather modest. Maybe you have two different sets of enzymes that actually can produce phosphatidic acid. So that remains to be checked in neurons as well.
And one more, you talked about catecholamines. So which catecholamines you're talking about?
It's adrenaline and noradrenaline, which is epinephrine and norepinephrine for Americans.
No, nothing to do with the dopamine or serotonergic system?
No, those cells are, dopamine is the precursor.
And the serotonin, which is very important in the,
Depression, yeah. Yeah, we haven't checked that, but it's a possibility that it's also working in this system.
Other question?
Yeah, so I have one. So it's fantastic. Couple things. First, I guess PLD was also a nominee for the George Costanza Award for a long time for lipid signaling. And maybe this question will show some evidence of cognitive decline. So all the steps that you actually lay out are also PIP2 dependent steps. And Tom Martin, of course, has shown that CAPS, which is probably involved in this, is PIP2 regulated for priming, and then endocytosis is PIP2 regulated. And then, of course, PLD1 is a PIP2 activated enzyme, and PA activates the Pi4P5 kinase that generates PIP2. So in your treatments, do you actually know what the PIP2 status is at the sites that you're looking at? The idea being that maybe PA is a fast forward for generating PIP2 pools, and that's really what you're measuring.
We have tried to address that, but measuring PIP2 levels precisely is not that easy. So one of the way to actually circumvent the possible limitation is what I said, is using a very narrow window to generate those lipids in order to prevent these long-term effects. Because I'm pretty sure that, affecting PLD will probably also affect PIP2 at some levels. That's for sure. So that's one of the way to actually to do that, is to provide the lipid in a short time and a specific location in cells. That's at least a step forward into the good direction. But I think it will be difficult, since those two are so intertwined, to have a very precise way to exclude.
Thank you.
Thank you.
Other questions? No, there's a chat by George Cobb. Oh, please, Dan.
Yeah,
Please.
I just have a quick comment. So Nick, that was a beautiful talk and very exciting, actually. Just a quick comment that Casey Barber in my lab does have evidence that PLD1 is probably involved in exocytosis in mammalian neurons as well. But the complication there is that the PLD is actually involved in neurons and in glial cells, and we're not quite sure what that communication's about. But that was a beautiful story, Nick. It was a great job.
Thank you.
Okay, thank you, Dan. Thank you again, Nick. It was Nicholas, very, very, very nice talk. And now is the time to move to the last talk by Laureen Parker-Jackson, Vanderbilt, is Understanding the Structure and Function of Tepsin in Autophagy. Please, Laureen.
Thank you. Good morning from Nashville, everybody. Thank you so much, Lucio, for the invitation. I'll echo Lois in saying, it's such a huge effort to organize a hybrid meeting, and we really appreciate being included. And someday I hope to meet you all in person, including Lucio. Until then, I will have to make do with, thank you, Lucio, for sending a little piece of Bologna to Nashville. We have an exhibit at our local art museum that arrives in a month. So I'll be there to appreciate a little more of your beautiful city, at least as far as we can from here. So my lab is really a structural and biochemistry lab, and we use these sorts of techniques to study mechanisms of membrane trafficking. And so the lab is broadly interested in coat protein complexes that drive formation of membrane-bound structures. So many of you will know these large multi-subunit complexes that drive many of these important events. And today I'll focus on the adapter protein or assembly polypeptide AP family that's related by evolution. So you're probably familiar with clathrin coats that include AP1 and AP2, but there are several members of this family, and some of them differ quite a bit from clathrin coats. And so today what I want to talk about is an unpublished story on the AP4 coat. And this is very much a story in progress, and I really welcome your thoughts and feedback on the multiple confusing aspects. So as far as we know, sort of when I started my lab a few years ago and how we think about the biology today, AP4 localizes here to the TGN. This is sort of the classical cell biology view, but many questions really remain about its structure and its function in cells. So I'll give you a brief overview for those of you unfamiliar with this coat. So here are some of the key proteins. AP4 itself is a tetramer of two large, a medium, and a small subunit, and it's thought to be recruited to the trans-Golgi network by ARF1 in its GTP-bound state. AP4 is an interesting one because it's been lost along the way over the course of evolution. So all of those useful model organisms that we'd love to use don't have AP4. So several years ago now, Scotty Robinson's lab identified tepsin, this protein I'm showing at the top of the slide, as a major component in these coated vesicles, and we sort of think of it as an accessory protein based on how we think of other coats being built. Now, in the last few years, a number of interesting cargoes and binding partners have emerged for AP4. So one of the cargoes that has been established by at least 3 labs now is Atg8, a protein involved in autophagy. It's an essential autophagy protein. Other cargoes include the syrinx 1 and 3, which are a little bit more mysterious. And very recently, as of now, just in a bioarchive paper, is diacylglycerol lipase beta, or DAG-LB, which is important for endocannabinoid signaling. So other interesting binding partners include Rusk2 and these hook proteins. And both of these proteins are thought to couple things to motors. So Rusks to kinesin and hook proteins form part of a larger complex that couple things to dynein and dynactin. So overall, it seems like the emerging role for AP4 in the cell is possibly a role in autophagy because it is needed to sort Atg8 out of the trans-Golgi network. And then even more recently, perhaps this emerging role in axonal maintenance through its role in sorting DAG-LB and then, of course, promoting endocannabinoid signaling. So although AP4 has been lost in model organisms, we've known for a long time it's important in humans. And that's because when it's lost in human patients, they have really terrible diseases, the spastic paraplegias, which are diseases of the peripheral nervous system. So one project we had, sort of during lockdown was a collaboration with my former neighbor, Tony Capra, who's now at UCSF. And so we thought about AP4 genetic variation and evolution with his lab and tried to do some prediction of pathogenicity in the context of structural model predictions. So, part of our work has been around providing some structural information to help medical doctors and families sort of understand these newly identified mutations and how they impact disease state. But for a long time now, part of my lab has focused on tepsin. So the first thing we did was to identify a binding motif here in the very C-terminus. Juan Bonificino's lab also identified this motif, and we showed that it interacts with the beta-4 subunit, the C-terminal part of AP4. And this mutations in either side impair the ability of tepsin to come to membranes. The other thing that we did was to determine structures, X-ray structures, very old-fashioned, I know, of the nth and VHS-like domains. And we got some surprises there. So our nth domain, classically nths are supposed to bind phospholipid head groups. Our nth domain does not have that traditional helix 0 or helix naught that helps form that lipid binding pocket. So that was a bit mysterious. And the VHS-like domain, again, looks similar to other VHS domains, but does not bind ubiquitin, as some trafficking VHS proteins do. So a lot of questions about what is it that tepsin actually does, since we sort of tried to test some of the canonical functions. So for a long time, I wondered whether tepsin was maybe a structural protein, because we've never identified, or as far as I know, no one has identified a scaffolding protein like clathrin in AP4 coats. But as this role for AP4 in sorting on autophagy protein emerged, I had a new student, Natalie Wallace, who joined the lab. And we started to look for these things called Lear motifs, or LC3 interacting region motifs, in different AP4 coat components. And so right here between our two structured domains, we found this little motif that was strongly predicted, whatever that means, to be a Lear motif. And basically it means that we've got a tryptophan and a leucine separated by two residues. And there are some amino acids are preferred at different positions, and that allows it to bind LC3 more strongly. So this WDEL is a strongly predicted motif. And these Lear motifs, for those of you like me who knew nothing about autophagy, they bind Atg8 proteins, which undertake a lot of different and sometimes I think conflicting roles during autophagy. So let me pause and say, I never envisioned working in autophagy. I'm a chemist by training, and it's a very terrifying and confusing field. Now, let me just remind you what Atg8 proteins look like. So there are 3 subfamilies in mammals. They contain, this is maybe important, to be determined. They contain a ubiquitin-like fold. So I'm showing you ubiquitin here on the right. And these guys each have that ubiquitin-like fold with an N-terminal extension. And what caught our attention was that VHS and nth domains actually bind ubiquitin in a lot of different proteins. So from a structural and evolutionary point of view, we could envision maybe how parts of tepsin could interact with this family of proteins. So Atg8 proteins can be either soluble or membrane attached. They become lipidated and attached to phosphoethanolamine through a conjugation cascade that's sort of analogous to the ubiquitin cascade. And depending on the different protein, they are implicated in different stages. So LC3 is really a key marker in autophagy and implicated in early stages of formation of the autophagosome, whereas these other families, GABA RAPs, seem to be implicated in later phases of autophagy, including the expansion or closure, or maybe even the fusion of the autophagosome with the lysosome. So we set out to test whether tepsin could bind this family, having identified a potential Lear motif. So here we're turning back to things I feel more comfortable with, whether or not full-length tepsin binds one member of each family. So here we have purified recombinant proteins expressed and purified from bacteria, and we're using GST pull-down assays, and here we have full-length tepsin as bait, pulling down his-tagged Atg8 proteins as prey. I don't know how clearly you can see it on the screen. There's kind of a weak band for LC3 here on our Kumasi stain gel, and you can see it more clearly when we blot against the his-tag on the Atg8 proteins down here at the bottom. It seems as though full-length tepsin prefers LC3 over the 2 other members of the family. In the lab, we also do a lot of calorimetry. So here we're trying to quantify binding of the tepsin motif to LC3B using calorimetry. There is a well-established binding pocket on LC3 circled here on the left that's known to interact with Lear motifs. So we tested whether wild type and mutant LC3 could bind this short tepsin motif in the calorimeter. So I'm showing you on the right just 2 representative isotherms. Of course, we do 3 or 4 runs total. And what we see is a dissociation constant around 40 micromolar, which is weak and in line with published data, because the thinking is that you would have lots of these motifs perhaps on a membrane. So then we wanted to come and think about how tepsin and LC3 interact in context of the full-length protein. So back to a bit more biochemistry, what we've done here is to introduce mutations either into the tepsin Lear motif or into LC3, and to see how that affects binding in our pull-down assay. So here in this sort of very middle lane, we've got wild-type tepsin and wild-type LC3B, and again, they seem to interact. Whereas if we have wild type tepsin and that mutant of LC3, we appear to lose the interaction biochemically. The same is true if we mutate the Lear motif and look at its interaction with wild type LC3. So far, all of these data sort of suggest that tepsin could have this kind of canonical Lear motif that binds in this established binding pocket. But as always in science and life, the biochemical data maybe to me don't fully add up yet. So we also thought about truncating our tepsin domains to see if we could get at what was really important for the interaction. And so if we start to do this and we test just the nth domain on its own, we don't see an interaction with LC3 at either Kinase or Western levels. However, if we include nth Lear, the VHS domain on its own, or the Lear motif and the VHS domain, then we start to see binding. So, we still wonder whether this VHS domain might be important for binding ubiquitin. Calorimetry has been complicated for technical reasons we can discuss if people are interested, but I still wonder if this VHS-like domain is somehow recognizing the ubiquitin-like fold in an analogous way. And then the point might be to, to promote a multivalent interaction and perhaps to increase avidity. Now, we noticed in our pull downs that when we have full-length tepsin, binding doesn't always appear as robust as when we start to cut off the C-terminus. So then we wondered if perhaps there was an auto-inhibited form of tepsin or something like this. And so to get at this in vitro, we decided to introduce AP4 beta-4 into the system. And to make a long story short, we don't think that that increases binding to LC3 at all, at least in vitro. So our next steps here are really to think more about maybe some liposome pelleting assays and try to understand what happens if we start to put things on membranes and if a clearer picture will emerge. But what Natalie really wanted to do when she joined the lab was to understand exactly what role tepsin is playing in cells as part of the coat. And this was very brave of her because those of you who know me know that I'm not a cell biologist. So she's really the first to focus specifically on the role of tepsin. And her very first glimpse is from a HAP1 cell line. So these are near haploid cells derived from a leukemia cell line. And she looked at HAP1 cells in which tepsin has been deleted using CRISPR. And in thin section EM, what we see, and I'll go ahead and say that we've already done this in different nutrient conditions. So it doesn't matter whether we look in basal or starvation conditions, we tend to see these large structures that look like they could be MVBs or autophagosomes that are enlarged in both basal and starved conditions. So we started to try to image these. It turned out they were very challenging to image. So then we moved into Hela cells, which are a little more established in the AP4 field anyway. And so we've done a number of things to measure and quantify endogenous LC3 and also to look at LAMP1 to try and get at the autophagosome, autolysosome situation in Hela cells. But what I want to share with you today was an idea that we took really from Lois from a talk I think she gave at Vanderbilt to try and establish if there's a phenotype using the autophagy flux assay. So again, for those of you who may not be as familiar, essentially what we have is a Hela cell line stably expressing a tandem reporter of LC3, RFP and GFP. And the idea is that in the early stages, when the phagophore is being formed and before it fuses with the lysosome, you will get yellow structures because you essentially can see both fluorophores. And then when autophagosomes fuse with lysosomes, the GFP gets quenched, and so things turn red. And so this seemed to give us a clearer phenotype, although, to me, it still seems kind of subtle. What we tend to see in either wild type Hela cells or in this line is a reproducible phenotype under basal nutrient conditions. So here I'm just showing you a glimpse of the data since I don't have enough time, but this is either tepsin or a non-targeting control siRNA in this tandem cell line. And across hundreds of cells, what Natalie sees is a reproducible increase in autophagosome number and volume under basal conditions when she knocks down tepsin transiently. She also sees a slight increase in autolysosome number. And again, we see this in the basal condition more so than in starvation conditions, serum starved conditions. So we, of course, also looked at AP4, since this is an important part of the AP4 coat, and it gives a slightly different phenotype, which we're still trying to untangle in our heads. We would have thought it would be purely additive because tepsin really depends on AP4. Here we see under basal conditions an increase in autophagosome and autolysosome number. But under starvation conditions, we also see a decrease in autophagosome number in AP4 depleted cells relative to control. So what Natalie is doing right now is really to look at an important cargo, Atg8, in these cells. And, her first pass is that she sees exactly what others have seen, that if you knock down AP4, Atg8 gets trapped in the Golgi. And so the next step is to try and figure out what happens with tepsin. We know that if you knock down tepsin, Atg8 still gets out. But then I think the question is, what happens to it next? So in my last couple of slides, I want to kind of summarize and then provide a model. And as I said, I'd love your feedback. So we think that we've shown that tepsin binds LC3 in vitro with micromolar affinity using kind of a canonical Lear motif that's pretty well documented for binding LC3 in the docking site. It may be that the VHS-like domain further provides another way for tepsin and LC3 to interact. Our most convincing data so far is that if we knock down tepsin in Helas, we see increased autophagosome number and size or volume under basal nutrient conditions, whereas AP4 depletion tends to have a slightly different effect. But I think this raises a lot of questions. The number one question to me is, why does tepsin bind LC3 and where in the cell does this happen? I wonder if these data with a lot of published AP4 data may suggest that AP4 coats help build an autophagosome more directly. And then finally, for those of you who are kind of experts in this field, there's this emerging consensus, right, that nutrient sensing is so important and drives trafficking. This is true for AP5 coats, for example, and I will not be surprised if this is what we find for AP4. So this is sort of the model that has emerged. I've borrowed this from one of Juan Bonificino's paper and adapted it a little bit with questions. So the thinking is still that AP4 coats form here at the Golgi, where they bind Atg8 as a cargo. Then perhaps by coupling to Rusk2, AP4 coated vesicles move in this anterograde direction. And I think people believe that they're helping to generate this Atg8 vesicle pool out here that's somehow important for forming the PAS. And I think one question is, do these vesicles remain coated with AP4? I think there's disagreement about that in the field. And if so, for how long? Because we could maybe envision that if they stay coated, that tepsin and LC3 and interaction there could help somehow build this structure. That's wild speculation. And then I think what's even more confusing is why AP4 binds, a kinesin and a dynein-dynactin adapter. And so Juan has proposed that actually maybe AP4 mediates trafficking in both directions, via kinesin this way and, coming back via the hook dynein-dynactin interaction to return things. But, why it does this and kind of how a vesicle would become recoded, I think that's still quite mysterious. So I want to finish by thanking those who bravely took on this project, which I think is interesting, even if it is confusing to us. I spoke a lot about Natalie. Our postdoc, John Gadberry, helped with some of the biochemistry. And I didn't really have time to talk about this today, but we have this amazing undergraduate, Olivia, who will hopefully be coming to AstraZeneca in Sweden. But she has knocked out AP4 genes in zebrafish and is now looking at some autophagy phenotypes there. And I hope to share those data with you soon. Thank you. Happy to take questions.
Thank you that much, Noreen. It's a very nice talk, which is now open for discussion. Questions here in the audience?
Hey, Lauren, that's John.
John.
Quick question. Yeah. I wondered, could you remind me on the AP4 mutations that occur in the disease states? Have you put those into in vitro systems to look to see whether they impair, and if so, what do you know about the functionality there?
Right. So we don't have access to these lines, but Scotty certainly does. And some of the folks who collaborate with her through Boston Children's have looked in patient fibroblasts. So there's kind of an exciting new paper from, his name is I've forgotten his surname, but Darius. And they have really established Atg8 getting trapped in the Golgi as a diagnostic test for these patients. What tends to happen in human patients is that they lose the whole complex. And so it is destabilized and Atg8 gets trapped there. So we do have some cell lines that we can use to look at that in the future. So far, we tried to get the undiagnosed disease network at Vanderbilt to look for any of these mysterious neurological diseases to see if we could identify either new AP4 patients or tepsin patients. And we have not been successful so far.
Okay, yes. No, first is Lois. Lois asked for. Ladies first.
So,
Lois?
Lauren, great talk. So interesting. And maybe I just got confused and everyone else has it straight. But I thought that if ATG9A would get trapped in the Golgi, then you should have less autophagosome formation.
That's what I know, Lois. That's exact. We have this conversation in lab meeting at least every quarter, because the phenotype that people see does not seem to match what we, or I mean, not what I understand about how an autophagosome should form.
So maybe part of it has to do with something that you explained to John York. So all those patient mutations. So
Yeah.
I heard one speak a week ago.
Okay.
And all those patient mutations, as you point out, destabilize the entire AP4 complex. And yours is different because it's impacting AP4 function, but differently, except you're still seeing ATG9 trapped in the 9A trapped in the Golgi. So what's ATG9B? Like, is there another ATG9 that could be?
So yeah, let me. I may have gone through this too fast. So if we knock down tepsin, so Natalie is still doing this, but both Scotty and Juan find if you knock down tepsin, ATG9 still gets out. So our idea is maybe, so if tepsin, the part tepsin plays is to help ATG9 get to the PAS, then it should become trapped at a different place. So that's the idea. I mean, she is really working on it as we speak. And so that may be harder to quantify. Looking at these peripheral pools, I don't know how successful we're going to be, but we're going to at least try.
Right, wonderful talk. So this might be a naive question, but in the patients that are affected, what cells are actually the ones that are really susceptible? And the question then is Hela actually the cell type that you even want to be looking, you know, at?
It's easy for me to answer that second question. No, it is not at all. You know, it was just because Hela cells are a little more tractable for us, especially because I had just started my lab. But the cells that are most affected undoubtedly are neurons. And I think the thinking for a long time has been that you really need autophagy to clear the neurons. And so perhaps, you know, one explanation for why the patients are so sick and deteriorate so rapidly is just that, you know, you're getting too much clogged junk in your neurons. Now, Alex Davies and Georg Borner's bioarchive paper point to a different and perhaps additive effect on this endocannabinoid signaling, because that really comes down to whether your axons are long enough and are being maintained. So anyway, you know, I think there's still some surprises in store for us all in terms of what AP4 does in cells, but neurons, yeah.
So that makes sense to me. So maybe an interesting assay in Hela cells would be to damage the mitochondria with a laser and then essentially ask what happens to clearance of the damaged organelle. There are actually established assays for this, and this might be an interesting look.
I wish I was there to have dinner with you. We have found a protein, a mitochondrial, well, maybe it's a mitochondrial protein that's supposed to interact with tepsin. And this has never made sense to me, but I might email you. Thank you.
Other questions?
Be careful of getting doing any kind of interactions with pancreatitis in person or via collaboration.
But put your mask back on, John.
Save your voice for later. Mina, last question. No. Lorraine, another question is coming. Wait.
Where would you place mTOR here in your pathway? Because suppression of that induces the autophagy?
I mean, it's an excellent question. We have tried, so let me back up and say AP5 is known to now pretty well established to depend on mTOR, and there seems to be an inverse relationship in terms of when the AP5 coat gets recruited to membranes. I would not be surprised if AP4 turns out to have a similar dependency. We tried to look at mTOR signaling using kind of that well-established antibody, and we were not successful in our first pass. So it's something we need to go back to and look at more carefully. You're right, it's an important point.
Okay, Thank you. No more questions. Thank you again, Lorena. It's nice talk. And now it's the final part of the symposium. Just a sec. Okay, here we are at the very final part of the symposium. Actually, you say, okay, this is the main spot. But I tell you this, that the guy who gave the special symposium lecture for many years, Sir Hans Krebs, he also always was used to say, this is just one part of the symposium, because the symposium itself is a very, very nice spot. So now, the special symposium lecture. This now is named to George Weber and Professor Manzoli. And look, this is an unbelievable list of speakers with the special symposium lecture. Many of them have been Nobel Prize. Some of them are fantastic scientists. There's only one very bad dot in 2000, but that doesn't matter. Okay, it's my usual joke every time. I beg your pardon for that. But I want to draw your attention also for another very interesting talk, the one by Paul Talalay in 1986. And this then, you know, the paper that came out from this talk is the paper with the highest citations in entire biomedical area. It's about 3,520 citations. I've got that paper by Paul Talalay. I know him, I knew him actually, and he was a very great person. So now we are just talking about the special symposium lecture, and this is a picture of the last talk, the last symposium who was attended by Sir Hans Krebs. And this is the symposium for the celebration of the 50 years of advances in enzyme regulation. And that time, the lecturer, special lecturer was Tim Hunt. And I tell you, I want to spend a few minutes. I tell you why he's here, not in the position usually just on the right of the chairman, because this is a Photoshop. He gave the talk for sure, but waiting for the picture, he was completely impossible to find him. You know where he was? In the cathedral, just in the middle of the cathedral, looking. He switched off the telephone, looking at the meridian, and he stayed there for more than half an hour looking for the huge meridian, which is in the center of the cathedral, San Petronio here in Bologna. And this is the other, sorry, I made something, okay. And this is the one that we had to celebrate the, this is wrong year, it's not the 50th, but the 60th symposium in Bologna. Actually, this was the last one in person. So this year we are in this mixed way, both in person as well as remote and virtual, but I do hope that next year we could meet all together, you, some of you, and new other entries, you know, it depending on, and could be also having the special lecture in this magnificent hall, which is absolutely fantastic. It's really great. And tonight, this is the lecture, special lecture is by John York. You already saw him. Many of you know him very well. Me too, as well as Vito. We know him ever since. And okay, Vito, don't look at me like that, you know. And it's a pleasure for me now to ask Professor Manzoli, our head of department, to introduce John for his special symposium lecture. Come here, Lucia, please.
Thank you, Lucio. It's a special pleasure for me to be here presenting the special symposium lecture of such a special symposium lecturer, which I'm so happy to introduce, even if it would not need an introduction. Dr. John York is a Midwest native with maternal Italian roots. He received his Bachelor of Science in Biochemistry from the University of Iowa, and his PhD in Molecular Cell Biology and Biochemistry from Washington University in St. Louis in 1993. Then research assistant, teaching assistant, postdoctoral fellow with his mentor, late Phil Magirus, who has been Spatial Symposium lecturer at the Advances in Biological Regulation Symposium in 2011 and 2013. From 2012 to 2020, John York was professor of biochemistry and the chair of the Department of Biochemistry at Vanderbilt University, where John's laboratory paved the way for discoveries in protein structure and function, cell signaling, and molecular genetics, and furthered our understanding of the pathophysiology of disease. Under his leadership, the Department of Biochemistry became 2019's top NIH-funded department in the nation, and nearly doubled the number of trainees. Before joining Vanderbilt, John was an investigator for the Howard Hughes Medical Institute and professor of pharmacology and biochemistry at Duke University. John York joined Impossible Foods in January 2021 as Chief Science Officer, and oversees the company's research and development and product innovation functions. His work focuses on building Impossible's technology platform, expanding basic research capabilities, accelerating next generation products, and recruiting the world's best scientists to join the Impossible Foods team. Throughout his career, in his laboratory, John trained over 50 scientists and over 200 graduate students, postdoctoral fellows, undergraduates, and high school students through thesis, departmental, and community activities. And to conclude, some friends told me that outside of the lab, he enjoys spending time with his family, off-piste skiing, playing tennis, woodworking, building outdoor pizza ovens, cooking, and more important, eating good Italian food, traveling, and reading the news from a variety of intellectually rigorous sources. And last but certainly not least, he's foreign corresponding member of the Academy of Sciences of Bologna. Thank you, John, for being here, please.
Wow. Can everybody hear me? Yeah. Thank you, Professor Menzeli. It's truly an honor to be here. I really wish, I'm going to get all teary-eyed. I wish I was there in person. My schedule just didn't allow it, but we have so many fond memories, my family and I, of being in Bologna and in Italy. It's also an honor because one of the first meetings that I attended was this meeting back when I started my career. So this is actually my, I think, 14th or 15th advances meeting. So to give this special lecture is truly an honor, and to have, connections to the University of Bologna and Lucio and such has just been a pleasure for me. And I want to thank the organizers, Lucio, and of course, the angels of Bologna, Julia, Roberta, Matilda, and Stefano, for for making this possible. So many trips. So Lauren, I hope you get there next year. I hope I get there next year as well. And we can properly walk up the hill together and share the sights of Bologna. All right, so enough yammering. I will start sharing a screen here. And sorry, let me just change and swap displays. Hang on 1 sec. Okay, so I titled this talk sort of a trip down memory lane because it may actually be one of the last talks I give about my academic work. And so, you know, obviously being in Bologna is far better than being in California. But here's the memory that I have from 2019 as we were gathering to come up to Lucio and Erminia's house, and he gave the blessing for us to arrive and such. So Lucio, you will always be a brother from a different mother, but we share our Italian roots, and for that I'm grateful. So my career actually, I think, as a scientist began in my basement, my parents' basement in 1972. My dad was kind of interested in chemistry and engineering, and so I was making hydrogen gas. So I think I must have been curious about science. I was curious about just about everything and how it worked. But I think this picture shows just where I was going at maybe an early age. I became enamored with molecular biology when I read a paper in high school in Scientific American about the recombinant DNA era. For the younger members of the audience, you know, these things called restriction enzymes came about, and the ability to snip DNA and actually make recombinant organisms was something that I found so intriguing. Possibly Bankitis, this is my best paper ever. I wrote it in my junior year of high school. I'll send it to you if you want to read it. It's quite illuminating. But it highlighted the fact that I think I really love the idea of using molecular approaches to solve problems. And, you know, shortly after graduating from college, Sally and I moved to Merck Labs, and I got a glimpse of what it was like to work on an applied project, but using discovery science. At the time, Roy Vagelos was running Merck, and it was a research-based company. So natural products were a very important component, and we studied vampire bats. And these vampire bats were interesting to Merck because of their saliva had powerful anticoagulants and agents that prevented clots from forming and breaking down clots. So I actually, the first gene I ever helped clone was a vampire bat plasminogen activator, which is now used, the knowledge that we gained from studying that protein is used in second, third, and fourth generations TPAs for patients that may have a heart attack. So it was really a great voyage for me in curiosity. Merck also was, in my mind, where I got energized by science. I mean, the place was amazing, and we, you know, had resources to do all kinds of different discovery science projects. And it's also what led to me meeting my mentor from grad school, Phil Majerus, who's, you know, I just label him in simple terms as chosen family. He's a great mentor. As Professor Manzoli mentioned, he gave the distinguished lecture. He's an unbelievable physician scientist and a true friend. And that was a picture from 1992 when I was in graduate school. My first attended meeting in advances in enzyme, I guess it was enzyme regulation back then. George Weber was hosting it in Indianapolis, 1997. The title of my talk was Nuclear Nostal Signaling and a Structural Functional Approach, which I think pretty much says it all in terms of what I've done. And then, of course, watching Lucio at Silvio's on my 12th advances meeting in 2019, not 2020, was truly special for Sally and I as well. All right, and as Lucia mentioned, I do have some Italian roots. Lucio and friends helped me actually go explore them. We took a trip to Sestola near Modena, and it's a very small farming community that is up in the mountains. And you know, my great grandfather Amo Bazzelli was from Sestola. My great grandmother Maria Pellini was from Belluno RCA, and they, you know, my dad was born in the States here, and then married my mom. So I'm truly grateful for my Italian heritage, and maybe someday I will get an Italian citizenship or passport. That would be fun. All right. And then I want to begin really by saying part of the talk is of thanking all the members of my lab from 1996 to 2021. My last student will graduate in about 3 weeks, and you know, it's been an incredible ride to have such talented people doing the work. And so this just shows a few pictures of them. So when I began my lab, I was really interested in trying to understand how cells communicate. And it was always very puzzling to me that if you did a literature search back in the 90s, you know, there were so many hits when you looked at inositol signaling, and just about everything activated the pathway. So this is typically an IP3 and a calcium-mediated pathway that was discussed then, but not only receptor tyrosine kinases, but G protein coupled receptors, such as GPRs. A photon of light can stimulate IP3 production. A cytokine and a growth factor can do the same. And it was intriguing to me that they all activated the PI signaling, so to speak, but yet they led to very, very different responses. And so, you know, we started thinking about this as a much more complex system than just the IP3 and calcium signaling axis. And we, for those that don't think about an inositol phosphate all the time, it's a 6-carbon ring. We sort of think of it as a rudimentary computer chip that has basically 3 states on each of the hydroxyls. You can either be unphosphorylated, phosphorylated once, a monophosphate, or even a diphosphate, as we've seen here at the meeting. If you think about the complexity of such a chemical scaffold, because of the asymmetry of the 2 position being axial, you can actually make 63 monophosphoester combinations and 728 mono and diphosphoester combinations. So overall, this is a massive amount of potential signaling for the cell to undertake. And we spent most of our career in the lab trying to understand how different inositol phosphates were made and how they regulated cellular events. And it was through the molecular approaches that we took that sort of helped us illuminate that. So how do you actually identify what an orphan molecule may do? Well, our approach was to go into a system that was genetically tractable and biochemically tractable. And so, you know, initially we went into yeast, but we've used just about every organism on the planet to try to understand how these signals might work. But if you take something away and then add it back in a very reductionist approach, you can understand what these molecules do. So, you know, I think my highlight of being interested in molecular biology came to fruition over my 25, 30 years of a career. These are some of the genes that we've spent time studying. It's not meant to be an exhaustive list, but for example, inositol polyphosphate 1 phosphatase is what I studied and cloned as a graduate student. And there's some lipid phosphatases on the list. There are some inositol kinases on the list. There are even things that are inhibited by lithium that have nothing to do with inositols that work on sulfation. And so through this journey, we were really able to use that enzyme regulation to help define new roles for inositides in cells. And so I thought I would just share a few of those as going down memory lane. And one of my favorites is when I cloned the INPP1 gene, or the 1 phosphatase in Phil's lab, we solved the structure by X-ray crystallography. And Lauren, back then, we actually used film. We had precession cameras to actually determine unit cell dimensions and space groups. It was a long task. I actually had to print out mini maps, which were pieces of overhead projector, film on a printer that you look at topographical maps. And I remember stacking them up with cardboard frames to make the Z-axis. And that's how I traced this model. And what you're seeing over here is the actual core structure of INPP1. And it turns out when we solved that structure, Merck had solved the structure of another inositol enzyme. And even though that they only shared a 6 amino acid sequence by homology, the protein structures were identical. And so that led to the discovery of not only where the lithium binds, these are all lithium inhibited phosphatases, but the lithium displaces one of the metals in the active site, which is required to inhibit that enzyme. The magnesium is required to activate it. And it's part of a family. And as the genomes became available, we searched with a more complex version of this motif based on the structure and found some new genes. And sort of as a bench to bedside, I mean, discovering using structural genomics, a family of proteins, and then studying, like, for example, this is a mouse knockout of GPAP that we made. It was a nucleotide phosphatase. It was a dwarf. It's one of the complementation groups for dwarfism or chondrodysplasia. And shortly after publishing this paper in PNAS in 2008, a physician in France actually had read the paper, shocking enough, and had a family of patients that were consanguineous progeny that had developmental defects that were mimicking our mouse knockout phenotype. And so they sequenced the patient's genome and they found that they were biallelic loss in the GPAP gene. So, while there's only a very few number of people, it does illustrate that by doing basic science, by following some curiosity-driven approaches, you can actually have an impact to understand the pathophysiology of human disease. As many of you know, when I began my lab, we were just looking at any gene we could possibly think of in the inositide pathway, and some of the interesting ones at the time were the inositol lipid phosphatases. And there were 4 such genes in the yeast genome as of 1996. And so we started doing basic biochemistry. And I bring this up because it's really kind of a fun story. We ordered some lipids to use in a biochemical assay. And it turned out they were actually not what was sold to us. They were the wrong lipids. We wanted PIP2, and I think they sent us PI. That there was some problem with that. So we got very frustrated, and we said, how can we do these assays to try to understand what lipid phosphatases might be doing without having radiochemicals to use? And so we came up with the idea. Xiuling Guo was the graduate student in the lab at the time. So we labeled inositol, inositol labeled yeast, which was a pretty common metabolic practice, not necessarily yeast, but just in general, and used that cellular extract to make a lipid micelles, micelles that you could then deacylate and run on an HPLC. And just showing on the top, you could see Pi, Pi3P, 4P. Those are the major monolipids in yeast. And then 3,5P2, we osmotically stimulated these cells. We also knocked out a phosphatase to elevate the levels of 4,5 so that we had more material. So we started going along, just doing recombinant assays, just taking our 5 phosphatases that we had as recombinant, ones that just had a 5 phosphatase domain, like INP5,4, or ones that look a lot like the protein that Pietro De Camilli had just cloned, called synaptogenin. And it was interesting because he named it synaptogenin for the god of 2 faces because of the Sac domain, which Vitas Bankaitis discovered on the suppressor screen, and also had a juxtapositioned 5 phosphatase domain. So when we gave it the 5 phosphatase and we saw that the lipid PIP2 was hydrolyzed and the product 4P was actually made, everything was fine. But where things got crazy is when we added the full-length INP5, 2, and 3, which is the synaptogenin ortholog, we noticed that all of the lipids disappeared. And I remember just saying to Xiuling, this has got to be an artifact. And thankfully, she told me to stuff it, and kept going with this and expressed not only the 5 phosphatase domain of synaptogenin-like proteins, but also the Sac domain. And look what happens when you just take that synaptogenin Sac domain. You don't touch Pi PIP2, but you hydrolyze these other lipids. And so that was really our first foray into a dual functional enzyme that had 2 lipid phosphatases that were encoded by different active sites. It illuminated why and some mechanisms by how SAC1 could be a second site suppressor of the secretory pathway mutant of SEC14. And it's still one of the most interesting things that you would put both of these lipid phosphatases on one protein. And the sequence, this CX5RT motif, was actually then used by a number of groups to find the P10, the 4-phosphatase family, and the myotubular proteins. And so, I think it was a great example of trying to use a combination of biochemistry and yeast to then understand some functionality. The other thing we were very interested in was phospholipase C, because at the time, most people thought you activated phospholipase C and you made IP3 and diacylglycerol, and that was the signaling axis. So we were just playing around, and we made a knockout of the only phospholipase C gene in yeast called PLC1. And when you radiolabel those yeast, you see a wild type yeast has mostly IP6, about 2% of the inositol label. Much of it is just free inositol. And there's a few different molecules in between. When we made this knockout of the null, we actually lost this. And when we overexpressed it on a very strong promoter, we actually could find that there was massive induction of all the inositol polyphosphate synthesized. And that led to the discovery of this phospholipase C dependent pathway, in which IP3 was actually now the precursor to making all of these other inositol phosphates. And that opened up a lot of really interesting collaborations. You saw a picture of Susan Wente, a very good friend of ours and colleague, who's now president of the Wake Forest University. Susan and I collaborated to actually help understand what these inositol phosphates might be doing. And lucky for us, these inositides were actually lipid derived in many organisms. And then different kinases that we cloned over the years actually were responsible for making those different inositol phosphates. And there's just a few of them are shown here. The kinases are conserved from yeast to man. They're found in just about every eukaryotic system. They have some deviations, but the core pathway to make IP6 is really something that is conserved. There are certainly some new areas that Adolfo has been working on that also indicate that you can make IP6 from a direct lipid independent path. That has been a really interesting discovery. We also wanted to make sure that we weren't just chasing the wind. So we looked to see whether these inositol phosphates were important in cellular and organismal function. And Andy Seeds knocked out one of the kinases, K2 or IPMK, in flies and found that it was a diskless phenotype. It was embryonic, or it was pupil lethal. And you can see that the fly adult body structures, here's a wild type that's emerged from the pupus. Here's one that is a knockout, and there are no body structures. In fact, you couldn't even pattern the imaginal disc that led to those. And then Josh Frederick also cloned and knocked out the mouse gene and found that it died at embryonic day 9.5. And so, it really became then a quest as to, well, all these things are happening. How are they regulated, and what are they actually doing in cells? And I just want to spend the last few minutes just sort of saying where we've gone since. And it's really been to try to decode the receptors for these. And I just show this for IP6 because an intriguing turn of events came about that really historically we should have paid more attention to. My actual undergraduate advisor, Arthur Arnone, who's a crystallographer who studied with the famous Max Perutz in the UK, had actually solved the structure of hemoglobin with IP6, or inositol hexaphosphate, bound. And how ironic was it for me years later to be actually studying IP6 in the context of cellular function. And a paper by Brenda Bass came out in 2005 that really caught our attention. And they showed the structure of this RNA editing enzyme, deaminase 8R2, actually structurally bound the IP6. And a graduate student in the lab found that if we took away IP6 and expressed this 8R in the cell, that it couldn't fold at all. So it was really confirmation that its functionality and its fold depended on an inositol phosphate, which is something we had never thought about. I mean, most IP messengers and lipids were these equilibrium allosteric type regulations where you had binding events or relocalization events, but it was exchangeable and reversible. In the case of 8R2, once the protein folds around this IP6, it doesn't let go. The only way it lets go is if it denatures. So we thought, well, you know, we've been searching for receptors of IP6 for, I don't know, a long time, genetic, biochemical approaches, didn't find anything. And it occurred to us, what if a lot of the other receptors to explain all these phenotypes we saw was due to structurally bound? And so Trang, Pham, and Jim Otto developed an assay to try to find structurally bound proteins. You can't just take IP6, radiolabel it, and add it to extracts and do receptor equilibrium binding assays. Instead, what we did is we used a mass-based assay for IP6 converted to IP7 with an enzyme, VIP1. And the assay is radioactive ATP, cold IP6, the enzyme, and you can produce in a linear fashion from 100 femtomoles to a picomole IP7 that you can then see as radioactive IP7 on a TLC. And the detection limit was pretty impressive. So looking at this sensitivity, and we could estimate that you could see about 100,000 molecules in yeast and about 12 million molecules in a Hela cell, just for perspective as to how much we thought, given the concentrations that we estimated at 20 micromolar. So that told us we could do a proteome-wide screen. And in order to prove the principle, Trang and Jim used ADAR as an example. And so this is a TLC that shows a standard where we just added enzyme, 0.1 picomoles. And if we added ADAR, which has an equal stoichiometric amount of IP6 bound to a mole of protein, if we added the VIP kinase to this, the ATP could, and the IP6 was so tightly bound to the ADAR that it never released and actually could be made into IP7, and you see very little IP7 produced in the ADAR protein added. But when you denature that ADAR before you actually add the VIP kinase shown here, so you denature it, you release the IP6, and now you add the radioactive ATP and the GAM and the VIP, you actually see now that production of IP7. So Trang and Jim did a proteome-wide screen, and I don't have time to go through all the details. This is all information that many of you have heard, but we uncovered a number of proteins that shared a common motif called the tetratricopeptide. There were a family of N-terminal acetyltransferases known as NAT A, E, and C, and then a protein of really mysterious function called clustered, or CLU1, which had a defect in mitochondrial function. So, we decided to take an approach to see whether or not the inositol phosphate was involved in the functionality. And Julie Neubauer and Jinhui Dong in the lab solved the structure of one of the N-terminal acetyltransferases called NAT-A. And this just shows the structure of the regulatory and the catalytic subunits, the blue and the orange. And the inositol phosphate actually binds along the interface of the two molecules and stabilizes that protein. And we've released all of these PDBs in that. And then if you look at the functionality, one of the most intriguing experiments I think we've ever done was to actually see if we could refold the activity of NAT-A in urea denaturation, the so-called Anfinsen experiment, and then test to see whether or not if we added back IP6 or an inositol phosphate, we could recapitulate folding. And to our pleasant surprise, when you actually add IP6 in a stoichiometric amount to the refolding buffer, you can take a protein that's 150 kilodaltons, 2 subunits, denature it such that it has no activity when you don't add IP6 back, and you get a robust activity when you do. So that suggested to us that in fact IP6 is functionally required for the fold of that protein and the stabilization. And we showed in cells that it's also required for the functionality. And what about CLU1? Well, CLU1 is also stabilized by IP6. And this just shows a Western blot of native CLU1 that we've tagged under an endogenous promoter in mutants that lack inositol phosphate. So IPK1, you can see, lacks just IP6. that protein is fully folded, and we now know that IP5 can substitute for IP6 in that. But if you knock out IPK2, where you don't make any of these higher inositol phosphates, you can see the protein is destabilized. And then in the PLC mutant, you can see there's no inositol phosphates, it's really destabilized. So we know that IP6 is also required in vivo and functionally for the CLU1 in its mitochondrial distribution. And I don't, again, have time to really summarize those findings. We solved the structure with a colleague at Vanderbilt, Yi Ren, who is a new expert in cryo-EM, and Yi and Bradley Clark, a student in my lab, collaborated to do the 3.4 angstrom resolution. They traced the entire structure from, this is a novel structure, it's a homodimer, and showed it with IP6. This is a figure that Yi made, and you can see there's a dimer structure, there's a ubiquitin-like domain, a dimer interface domain, and then the IP6 binds in those helical repeats. And so we're seeing a pattern emerge that these helical repeats have charged residues, lysines, arginines, and others to coordinate that binding. But that protein is actually, you can now see why it's structurally dependent on the IP6 being present. And if you look in a little bit closer, you can see just how beautifully coordinated by many, many residues that IP6 is. All right, so I just want to end there by saying that, by taking this approach, an enzymatic approach, we've been able to identify a number of different ideas and patterns in inositide signaling. The latest of which is how these proteins or these molecules can function not only as an allosteric regulator, but as a regulator of protein folding. And given that the tetratricopeptide family is an enormous family, there are hundreds, if not thousands, of these repeat proteins in the human genome, we also asked the question, were there any others? And so one of the things that happened just shortly after we got those results is Marco Sui, a postdoc in the lab, had been studying the phenotype of the fly knockout IPK2, and he noticed that they had a chromosomal sister chromatid separation defect, and that the cells that resulted were aneuploid. And you can see here that these are metaphase chromosome squashes. Here's a normal X shape of a chromosome that's been it's chromatid. And you can see in the IPK2 knockout that very few of these have still remain tied together through that cohesion complex. And then if we put in an active form of a plant enzyme, we can completely restore that. So it's really the product of this IPK2 that was important. And about, I don't know, a few years back, a paper came out in Molecular Cell that shows the basis for how a sister chromatid defect could happen. And it's one of the the heat repeat proteins that's part of the cohesion complex called PDS5, actually binds IP6 through these helical repeats and actually stabilizes that protein. So in all, there are lots of different proteins, we think, in the cell that bind these inositol phosphates. If we count up how many molecules there might be in yeast, for example, of IP6, let's estimate there's 100,000 molecules. There's 17,000 molecules of NAT-A, 5,000 of NAT-C, 5,000 of NAT-E, 10,000 of CLU1, 5,000 of PDS, not sure about 5,000 of ADAR. So, we're approaching over half of the inositol phosphate, IP6, is being protein bound. And I presented this, and Bob Michel, with an incredible photographic memory as well as just incredible contributions, said, well, we published this paper that showed when we permeabilized cells and we looked at release of inositols and various molecules, we found that IP6 and IP5 were actually retained by the cell in these. So that really is indicative when you do this tiny permeabilization by a toxin, that a lot of that IP6 in vivo is bound to proteins. So we think that this is the family. It's probably just the tip of the iceberg. And I think it's going to be a very interesting next decade to see how many proteins actually bind IPs, maybe even some lipids do the same. So, as I've transitioned now, I am trying to understand and use biochemistry to create plant-based foods at Impossible. And I'll just leave you with a statistic. There are 1.7 billion cows on the planet. In order to keep them alive, we use 45% of the Earth's ice-free land surface. And our mission at Impossible is to restore global biodiversity and avert a climate catastrophe by replacing the use of animals for food. So as many of you know, I am possibly one of the biggest foodies that's ever attended this meeting. As Professor Manzoli indicated, I love Italian food, but I'm hoping at some point to help produce a plant-based food that convinces all of you that it's not a sustainable future. So it's been an honor and a privilege for me to be here. Thank you so much for hosting this meeting, Lucio, over the years, and to the University of Bologna, Professor Manzoli. You know, you're just an incredible leader at the school. And so thank you all for your attention, and hope to see you again in person soon.
Thank you, John. Thank you so much for sharing with us a big part of your personal and professional history and pathway, which is also corresponded to 3 decades of history of sciences. And many compliments for this outstanding presentation, which confirms once again the clarity of your thoughts with ideas always moving forward. You have just answered to all my questions, but maybe one is if you could start again now, from where would you start?
Yeah, I think if I were starting right now with all of the, can you still hear me? My earbud went out. Okay. You know, if I were to start my lab right now, I would take advantage of the incredible ability to edit a single nucleotide of a gene, and I probably would go into something along the lines of neuroscience, because you know the frontier for the next generation is really to understand the neural network and how the brain functions, and I think that science is the techniques and the methods that we have available today. As exciting as it was for me to be able to use restriction enzymes when I first started, the ability to edit a single nucleotide out of 6 billion in the genetic code, and to think that that genetic code is replicated 1 trillion times to make a human organism, to me, that's just one of the most mind-blowing frontiers. And so I hope we use it responsibly in the future. And if you are interested in any good book reads, Codebreaker, or about Jennifer Doudna's journey, written by Walter Isaacson. And she also wrote a book, her first book, which was A Crack in Creation, both of which are really interesting journeys about how they got to that stage. So that's where I would go at this point.
Thank you.
And of course, inositides are critically important in that. So I would have eventually ended up right back to lipids and inositol phosphates.
Okay. Thank you, John. Is there any question here, or Vitas?
Well, John, you know, what can I say? I mean, this is emotional, I guess. So I always learn something new. Okay, and so I actually have a scientific question. So you showed that the PLC1 knockout, you completely destabilized the IP6 receptor. I forget which one it was.
CLU1, yeah.
CLU1. Why doesn't the lipid-independent pathway for generating inositol polyphosphates compensate for that?
Well, there is no lipid-independent pathway
In
that's
yeast.
been described in Cerevisiae, and we use Cerevisiae for that. So I think if we would have put that, it would be a great biosensor to see what organismal cells. So Adolfo, there you go. Just take clue 1 and express it in and see if it gets destabilized. And if it doesn't, you know, then you know that the independent pathway is really functioning to do that. So
yeah,
it's a
it
good
seems
question.
to me that actually tells you really what the contributions are. Right. Thank you.
Yep.
Thank you. Lois, I've read. Please, Lois.
So I would like to say something emotional and then ask a question.
No ballroom dancing.
I'll try not to cry. So John and also Vitas really welcomed me to the field when I was a vulnerable new scientist and couldn't get funded. They really encouraged me, and I'm going to miss that because I still need help. So Vitas, you can't just go do something wild and wacky and start working on something totally different. And John, I may still be reaching out to you with scientific questions. So thank you. So my question for you is, do you think that there's communication between the structural IP6 and the signaling IP6 that's going to now go to IP7 and all these other forms?
I think that's you know, I guess Professor Menzoli, perhaps what I would do next if my lab were still running is I would try to find out if that axis does exist, because think about it, you make an enormous amount of IP6 that you store in proteins in a cell. So if you were to degrade those proteins, for example, you would release IP6 in what would be perceived as a phospholipase C or lipid-independent pathway. And so, you know, we know that if we overexpress proteins that take IP6 to IP7 in vivo, that they don't just do that, indicating that there is a very tight regulation in that. And we published a paper where we could sort of sledgehammer it by activating a G protein and expressing these kinases, and we could get it to work. But it really does say that if you want to make IP6 or IP7 and 8, that there must be a pool, either de novo synthesized or a pool of IP6 that is destroyed, that is protein bound, that is then released after the protein is destroyed. And I think that that would be a very interesting concept, because then you could have sort of a a potential energy form of the IP6 sort of just waiting at the top of the mountain. You destroy what's holding it, and now you release it, and IP7 and IP8 can do their thing. And they clearly, IP7 and 8 have very, very important functions that a number of the labs have published over the years. And so for me, that's a really interesting idea. I would say that's a good place to go next. And don't worry, we'll make sure that you know, there's still plenty of support. I'm going to try to recruit Bankitis to Impossible Foods, but I don't know if we could take him. There's some people here that are pretty you know, beat to their own drum types, and I think Vito, you would fit in, all except for the fact that it's really hard to quit eating meat.
Now we are reading a chat.
John, there's no way you could be my boss. That's all there is to it.
No,
so did you have to become vegetarian? Did you have to become vegetarian?
I didn't have to. I've chosen to. So yeah.
Yes, actually, Adolfo is supporting with chat your tools.
Yeah, so I see this comment, plant-based food might contain a lot of IP6, isn't it ironic? Yeah, that is true. I can tell you a lot about it, but then I'd have to kill you, Adolfo. But believe me, IP6, back in, if you read some of Victor Raboy's papers, the literature about phytic acid, it's one of the most abundant molecules in seed. It's also chelating a bunch of micronutrients like iron and metals. So it's widely perceived as a worldwide health problem that many of the plant-based diets actually have too much phytic acid. And so there's certainly some very interesting ideas as to how to overcome that. And who would have thought that I joined a company and that I might even be able to contribute something scientifically.
Okay. So at the very end, as a bottom line, we can eat rice. So that's a good thing, yeah? Adding some
pasta too. Well, pasta, it's got some egg in it, but
adding some hot pepper, you know, now the hot pepper is, you know, in some ways got the Nobel Prize with Julius, you know, with capsaicin.
Yeah.
And that's it. And I want to thank you again. And before the final acknowledgement, I also should say that we are very, very proud of this moment here in Italy, even though not as biologists, but we got again a Nobel Prize in physics with Parisi. And actually it's something very, very important for the Italian scientific community. A few hours ago, yeah. Okay, thank you, John, again. Thank you, thank you. Okay, right. So we are at the end of the symposium. And as usual, the final acknowledgements. First of all, I have to thank all of you and all the speakers for the wonderful, wonderful talks. A special thank to John, but also I have to thank the colleagues here in person, Vitas, Ohad, that now is following us from Zurich, and Mina and Nicholas for chairing the session. You know, it's not that easy to run a so-called mixed conference. It's not that easy. Anyway, I think that we did well. And as you can see here, I have to thank the Academy of Science here in Bologna to host us in this magnificent, really magnificent location. Elsevier for the book that is coming out from the symposium, and just a very gentle reminder about October 15, because it's deadline for manuscripts. You know, I'm always pushed by this Elsevier person. So, you know, I'm just in the middle. It's very complicated for me. And I have to thank the Department of Biomedical Science, University of Bologna, and the head of the department, my friend Lucia, for all the support. But a special thank you to the Fondazione Cassa di Risparmio in Bologna that gave us the possibility since 2005 to run this meeting with a very, very, very generous financial support. And before saying the final word, the symposium is adjourned, I have also to thank my family that stand me during the time of the preparation of the symposium, and my wife Herminia, that she always tried to help us in the overall organization of the symposium. And what George Weber was used to call the silent angels, the people in my lab that very silently help in every moment. So Stefano, Matilda, Julia, Roberta, they are usually doing what I say, the work of the silent angels. Okay, thank you. Thank you again so much for being here for this mixed but still partially in-person event. It's a pleasure for me, and I hope that next year we can reconvene here in Bologna in person and really trying to enjoy to talk each other without any mask, without anything. I don't know. It's, you know, I always say I have a dream. Could be, could be. Thank you, and symposium is adjourned.